Hello and welcome to The Sleepy Scientist. Tonight's topic is biology, a calm glide from tiny cells to whole ecosystems, from quiet chemistry to living patterns. Where are you listening from tonight? If this helps you wind down, a like and a subscribe would be truly appreciated, and to everyone already subscribed, thank you. Please tap the notifications bell so the next gentle upload finds you right on time. May this be just intriguing enough to slow your thoughts, and if it keeps you awake a little longer, let it be for a good reason, leaving with one more small wonder than you arrived with. To begin, a soft question. How does something as small as a cell keep something as large as life in balance? Tonight's journey moves through life's essentials with a steady, quiet pace. The first stirrings in water, membranes that hold order together, energy captured and shared, genes that remember, and the partnerships that stitch organisms into communities. Patterns repeat across scales, branching, spirals, rhythms, linking lungs to trees, reefs to cities of tiny builders. No rush, no noise, just clear, simple steps through the living world so your mind can settle while curiosity stays warm. Life speaks with a small alphabet. 4 chemical letters: adenine, thymine, guanine, cytosine, pair like careful partners, A with T, G with C, making a ladder that twists into the familiar double helix. The elegance is in the thrift. 3-letter words called codons, written from those 4 symbols, map to 20 common amino acids. From that set, cells build proteins that act as pumps, scaffolds, switches, and engines. A slight change in a single letter can shift a meaning, sometimes barely at all, sometimes quite a lot. Yet across the planet the dictionary is almost the same. A bacterium in a hot spring, a fern in a shaded ravine, and a neuron behind a human eye all read the same code with the same basic translation table. This near-universal script hints at a shared ancestors deep in time, one lineage giving rise to many forms, yet carrying the same rules for reading life sentences. There are a few small dialects at life's margins, mitochondria and certain microbes bend a codon or two, but the message holds: one language, many accents. Watch the code at work inside any living cell, and the choreography is surprisingly tidy. Enzymes unzip the double helix and draft a working copy called RNA, which swaps thymine for uracil and carries instructions outward. Ribosomes, tiny molecular workbenches, read the RNA 3 letters at a time, clicking amino acids together in a chain. In water, that chain folds into a particular shape, and shape is destiny. Some folds make tunnels that shuttle sugars. Some form pincers that grab molecules and catalyze reactions. Some assemble like Lego pieces to build larger machines. It is a quiet miracle of combinatorics. 4 letters make 20 building blocks, and 20 building blocks make tens of thousands of tools, all arising from the same simple script. The stage that first let those letters begin to matter was almost certainly wet. Early Earth wore a global ocean, salted with minerals and warmed from within. Along spreading ridges, alkaline hydrothermal vents bubbled hydrogen-rich fluids into colder seawater. As those fluids met the ocean, minerals precipitated into chimney-like mounds riddled with microscopic pores. Across the thin walls of those pores, natural gradients formed, differences in acidity and chemical energy, much like the gradients living cells use today across membranes. Iron and sulfur compounds at those boundaries can catalyze reactions that bind carbon atoms together, assembling simple organic molecules. In settings like these, chemistry does not need a spark of lightning or a crowded laboratory bench. The rock itself provides structure, energy, and a steady trickle of ingredients. From simple molecules, complexity can creep in. Short strands of RNA are capable of 2 jobs: storing information and speeding up reactions. that dual talent suggests a plausible early chapter where small self-copying RNAs competed and cooperated inside mineral pores, some persisting better than others. Over time, fatty molecules, abundant in such environments, could have gathered into fragile bubbles. Those bubbles trap useful molecules, concentrate reactions, and split into daughter bubbles with a nudge. The world inside a pore begins to resemble the world inside a primitive cell. Later, DNA, which is more stable than RNA, may have taken over long-term information storage, while proteins, with their dizzying variety of shapes, took over most of the heavy lifting. The exact routes remain debated, but the outline is consistent. Water-rich settings, natural compartments, steady gradients, and chemistry learning to copy itself. Water is not just a backdrop here, it is an active participant. A water molecule is bent and polar, with a slight negative side and a slight positive side. Those tiny imbalances let water form fleeting hydrogen bonds, a handshake that lasts a moment and then rearranges, creating a liquid that can dissolve salts and sugars, ferry ions, and allow molecules to meet and react. Water's polarity pushes oily parts of molecules together, a phenomenon called the hydrophobic effect, and that push is what helps proteins fold and membranes self-assemble. The same property tucks the fatty tails of lipids away from water and leaves their charged heads facing out, producing stable bubbles that become cell membranes. Without this gentle sorting, the architecture of life would collapse. Water also buffers change. It has a high heat capacity, soaking up energy without wild temperature swings, which stabilizes environments from tide pools to bloodstreams. It is densest just above freezing, so ice floats, insulating lakes and oceans and protecting the life beneath. It even splits very slightly into ions, hydrogen and hydroxide, giving solutions their acidity and providing a quiet electrical background for the chemistry of metabolism. In cells, streams of protons flow across membranes, and that flow turns a rotary protein called ATP synthase, which snaps phosphate groups onto ADP to make ATP, the small energy coin spent by everything from muscle fibers to molecular pumps. The whole arrangement is so elegant it appears in bacteria, in the chloroplasts of plants, and in the mitochondria in human cells. Proton gradients power life because water makes those gradients possible. Step back and the pattern becomes simple. Water enabled chemistry. Chemistry learned to store patterns. Patterns learned to copy. Copying allowed selection. And selection, over deep time, wrote the diversity visible today. That shared alphabet shining through all living things is the fossil of this history. It is the reason a gene from a jellyfish can make a mouse cell glow green, or a bacterial enzyme can be borrowed to edit a plant genome. Compatibility across life's kingdoms is not an accident, it is heritage. This unity does not erase difference. A codon table is universal enough to bind life together, yet flexible enough to allow novelty. Redundancy in the code, multiple codons mapping to the same amino acid, acts like a cushion against errors. Slight shifts in timing or expression create new traits without breaking the whole system. Over long spans, such tweaks accumulate into fins, fronds, feathers, and fingers. Still, beneath all that variety, the letters, the words, and the way ribosomes read them remain familiar. Even the exceptions prove the rule. When a microbe uses a codon differently, it stands out precisely because almost everything else does not. In practical terms, this shared language lets ideas and tools flow freely across biology. Medicine can ask a bacterium to grow a human insulin protein. Ecology can track genes in seawater to count invisible microbes. Agriculture can nudge a plant to resist a pest by rerouting a pathway conserved back to the earlier cells. The lab bench works because oceans once did. In the end, the dependence on liquid water circles back to daily life in quiet ways. A sip from a glass sets ions moving across membrane channels that were sculpted by evolution to work in a watery world. A heartbeat is the coordinated action of proteins folded by water and powered by gradients built in water. A thought is a dance of charged particles through channels that open and close in a fluid environment. When the air is dry, lungs humidify it. When the day is hot, sweat cools skin. When cold threatens, blood shifts and water's thermal properties help hold heat. Biology never left the ocean entirely. It brought the ocean inside, within cells, within tissues, within the delicate chemistry that keeps a body alive. So the story begins with 4 letters, written in a solvent that makes their meanings possible. The alphabet is small, the library immense, and the librarian is water, arranging shelves and guiding readers without a word. Everything begins small. Before a body's a body, it is a single cell, a round droplet of chemistry wrapped in a thin membrane, carrying a set of instructions and the tools to read them. That first cell divides, and divides again, not as a frantic explosion, but as a careful unfolding of a plan. With each split, new decisions appear. Some descendants will learn to contract like muscle, some to carry signals like nerve, some to ferry oxygen, some to become bone and give shape. What seems like magic is choreography, genes switching on and off at the right moments, proteins guiding neighbours to their places, membranes bending and pinching to sculpt tissues. The result, after countless rounds of division, is a living architecture that can breathe and heal and think. Yet in spirit it remains a community of cells, each one tiny and alive in its own right. Inside an adult human there are roughly 37 trillion of these units, a number so large it is easier to feel than to picture. Imagine the population of a city, then of a continent, then of many planets worth of people, all working side by side. Each cell is a little world with borders, power lines, factories, roads, trash collection, and security gates. Some are spherical, some flat, some branching like trees. A red blood cell is a flexible disc built to slip through the narrowest capillaries. An intestinal cell grows finger-like folds to absorb more nutrients. A neuron stretches astonishing distances, a single cell forming a cable that may run from the spine to a toe. The diversity of shapes follows the diversity of jobs. Even the quiet places are busy. Skin is renewing, bone is remodeling, the gut is trading molecules with food and microbes, and the liver is sorting, filtering and filing a constant stream of chemicals. Walk through one cell and the city analogy becomes hard to resist. Energy comes from tiny structures called mitochondria, which act like power plants, breaking down fuel and charging the cell's batteries. The rough endoplasmic reticulum is a factory floor dotted with ribosomes, where proteins are assembled letter by letter from the RNA instructions copied out of DNA. The smooth endoplasmic reticulum handles oils and hormones, refining them for use or shipment. The Golgi apparatus stacks packages and adds address labels so proteins reach the correct destinations. Vesicles, small membrane bubbles, carry cargo along interior roads, which are really protein rails called microtubules and actin filaments. Molecular motors step along these rails in a hand-over-hand gait, hauling their loads with a determination that looks almost animated when seen under a microscope. At intersections, signals tell traffic when to go and when to stop, ensuring the right materials arrive precisely where they are needed. Recycling is a full-time job. Broken proteins are tagged and fed into the proteasome, a barrel-shaped machine that chews them into pieces for reuse. Bigger items, damaged mitochondria, invading bacteria, or clumps of debris, are wrapped into sacs and delivered to lysosomes, where potent enzymes break them down. This process, autophagy, is both spring cleaning and survival strategy. In lean times, a cell can dismantle non-essential parts to stay alive. Quality control watches everything. If DNA is damaged, sensors pause the cell cycle to allow repairs. If the damage is too severe, the cell may perform programmed self-destruction for the good of the whole organism, a quiet exit that prevents harm. At the border, the cell membrane is more than a skin. It is a gate that selects what enters and leaves, a conversation surface where receptors listen to hormones and growth factors, and a switchboard that converts outside messages into inside actions. Channels open and close to let ions flow, creating small electrical currents. In muscle, those currents trigger contraction. In neurons, they carry thoughts. In immune cells, signals from the membrane guide the decision to attack or stand down. Neighbouring cells talk by exchanging chemicals, but they also touch, connecting through junctions that pass molecules or anchor tissues. In a heartbeat, a wave of ions jumps from cell to cell, allowing millions of muscle fibres to work as one. Not every cell lives the same length of time. Some are sprinters, living fast and disappearing quickly. Neutrophils, a type of white blood cell that rushes to the site of a cut or infection, may work for only a day or two before they are spent. The lining of the small intestine turns over in just a few days, shedding old cells and replacing them with fresh ones grown from stem cells tucked into crypts. Skin cells migrate outward over weeks, forming a protective layer that flakes away as new cells push up from below. Red blood cells, stripped of their nucleus to make room for oxygen-carrying hemoglobin, circulate for around 4 months before being recycled in the spleen. Other cells are marathoners. Bone cells called osteocytes can live for years, monitoring the stresses placed on the skeleton, directing repairs. Liver cells are patient and resilient, dividing when needed and even regenerating large portions of the organ after injury. And neurons, especially in the brain's cortex, can last a lifetime, forming memories and habits that may outlive the cells around them many times over. Even here, there are nuances. Some regions of the adult brain do produce new neurons, and glial cells support and modulate neural circuits. Longevity depends on both job and environment, on exposure to stress, and on the balance between damage and repair. The shared language that lets all these different cells coordinate is molecular. DNA inside the nucleus stores the instructions. RNA carries them outward. Proteins carry them out. Signaling pathways with names like MEAP kinase or PI3 kinase turn messages into action, turning on genes, opening channels and moving organelles. The cytoskeleton, the internal framework, can be rearranged in seconds to let a cell crawl, divide, or change shape. In development, gradients of signaling molecules drift across tissues, and cells read their position by measuring concentration, like travellers reading distance by the dimness of a lighthouse. That is how a simple sheet of cells folds into a tube, pinches into chambers, and becomes a beating heart. Constraints guide creativity. A cell is small not by accident but by need. Diffusion, the passive drift of molecules, works best across short distances. That is why cells use networks of rails and motors for long hauls, why they pack enzymes close to their substrates, and why they fold their membranes into complex shapes to increase surface area. The same principles shape whole organs. Lungs branch and branch again to bring air within a breath of every blood vessel. The gut coils and sprouts folds to expose food to more absorbing cells. The kidney filters blood through countless tiny loops that concentrate wastes with minimal energy. Each organ is a community project, but the logic starts at the cellular scale. Even single-celled organisms show the full toolkit. A solitary bacterium senses food, swims with a rotating flagellum, and can exchange genes with neighbours through a fine tube. A single-celled eukaryote may hunt, flee, build a shell, or form a temporary colony. When the environment turns harsh, some cells incist or sporulate, sealing themselves off until conditions improve. The line between one cell and many cells is not a wall but a continuum of strategies for staying alive. Multicellularity evolved multiple times, and each time the key was cooperation. Cells specializing and sharing work, surrendering a little autonomy for the benefits of belonging. For all their independence, cells remember their membership. An immune cell that learns to tolerate the body's own molecules prevents autoimmunity. A skin cell that touches its neighbours knows it is part of a barrier and behaves differently than it would alone. Cancer, in this light, is less an invasion from outside than a breakdown of the social contract. A cell ignoring signals, refusing to die when damaged, hoarding resources, and migrating where it should not. Treatments, whether surgery, radiation, or targeted drugs, aim to restore the balance by removing or re-educating the rule breakers. And still, the wonder is in the everyday. A fingertip heals after a scrape because cells sense a gap, crawl into it, and knit new skin. A memory forms because neurons strengthen certain synapses and loosen others, a microscopic remodeling that changes how signals flow. A meal becomes part of a body because intestinal cells harvest sugars and amino acids, pass them to the blood, and the liver sorts them with tireless patience. None of these moments requires a command center barking orders. Local rules, followed well, create global order. That is the quiet genius of cellular life. Look closely enough, and the enormity of 37 trillion becomes friendly. Every beat of the heart, every blink, every quiet breath is a group effort by tiny citizens built from the same materials and following the same guidebook. They live short or long, simple or specialized lives, and they speak a language so old and widely shared that a cell from a plant, a fungus, or an animal would still find parts of the conversation familiar. From a single droplet at the beginning to the bustling metropolis of a mature body, the story stays the same. Small units, clear rules, careful cooperation, and from that, a living whole. A living cell is defined as much by its edge as by its contents. That edge is a membrane, a glimmering film 2 molecules thick, so thin that thousands stacked together would be narrower than a sheet of paper. Its ingredients are amphipathic lipids, each with a water-loving head and a water-avoiding tail. Drop them into water and they self-assemble into a bilayer, as if following a recipe. Heads out, tails tucked away. This simple geometry creates a boundary that is both sturdy and flexible. A velvet rope that lets the right guests in while gently keeping the wrong ones out. Selectivity is the first miracle. Water and a few small gases slip through with ease. Larger or charged molecules face a polite interrogation at the door. Channels, barrel-shaped proteins, form tiny tunnels that admit ions like sodium, potassium, calcium, and chloride. Pumps use energy to move cargo against gradients, stocking the cell's shelves with the molecules it needs. Transporters trade one substance for another, like shopkeepers with a knack for fair exchange. Together, these proteins turn a bland barrier into a bustling checkpoint. Because ions are separated across this thin film, the membrane becomes a battery. The inside of most cells rests at a small negative voltage compared to the outside, maintained by teams of pumps and leak channels. Nudge that voltage and channels respond, some open when they sense a change in charge, some when a chemical messenger docks, some when the membrane is stretched. The result is electricity on a tiny scale, spikes and waves of voltage that carry information. In neurons, a quick rise and fall called an action potential races along the membrane, leaping from one gap in the insulation to the next in a relay that can reach well over 100 km per hour. The signal ends at a synapse, where the electrical message becomes chemical for a breath, then electrical again on the other side. Thought and motion begin as brief flickers along a film only a few nanometers thick. The same physics powers muscles. A nerve impulse arriving at a muscle cell opens channels, calcium floods in, and proteins inside the cell slide past one another like rows of oars, shortening the fiber. Open and close, flood and clear. The sequence repeats, and a heartbeat or a stride emerges from the choreography. In hearing, hair cells in the inner ear wear tiny bristles. Sound bends them. Tension changes. Channels open, and the membrane translates vibration into voltage. In vision, photons strike membrane-bound opsins in retinal cells, setting off cascades that change the flow of ions. Darkness and light become patterns of charge that the brain can interpret. Sensing, moving, thinking, all of it starts with membranes that can feel and respond. Energy production also lives at the border. In mitochondria and in the green chloroplasts of plants, teams of proteins use membranes to stockpile protons on one side, creating a steep gradient, like water behind a dam. A rotary enzyme called ATP synthase allows those protons to flow back, turning like a turbine and clicking together molecules of ATP, the cell's spendable energy. Bacteria run variations of the same trick on their outer membranes and even power their flagella, tiny propellers directly from the flow. At the scale of life's engines, the membrane is not just a wall, it is the machine housing. Fluidity is the secret that lets everything adapt. Lipids are not cemented in place. They drift like people mingling at a quiet party, and proteins move among them. Cholesterol slips between tails to keep the film from becoming too stiff in the cold or too floppy in the heat. Cells can adjust the blend of their lipids to match the season. More unsaturated tails for flexibility in winter, more saturated ones for stability in summer. Islands of particular lipids and proteins, rafts, form and disperse, collecting the right players for a signaling event and then dissolving when the job is done. The membrane edits itself in real time. Curvature adds another dimension. To swallow something from the outside world, the surface dimples inward, coats itself with clathrin like a temporary suit of armour, pinches off and brings a bubble of cargo inside. To send something out, vesicles loaded in the cell's interior fuse with the membrane and spill their contents, neurotransmitters, hormones, antibodies, into the space beyond. Specialized proteins called SNARES entwine like zip ties to bring membranes close enough to merge. Every second, a cell performs thousands of these fusions and fissions without leaking so much as a drop of cytoplasm. Not all membranes are alike. The myelin that wraps many nerve fibers is a multi-layered sheath, fat-rich and insulating, that speeds electrical conduction by forcing signals to jump between exposed nodes. The membranes of lysosomes are reinforced to contain potent digestive enzymes. Plant cells surround their membranes with a sturdy cell wall of cellulose, a lattice that protects without blocking the subtle dance of ions and signals at the true living boundary within. Even viruses, the minimalist travellers at the edge of life, sometimes borrow a host membrane on their way out, wearing it like a disguise to slip into the next cell. Communication thrives on the surface. Receptors wait with outward-facing hands for growth factors, hormones and immune messengers. A single binding event can set off a cascade inside. Enzymes switch on, genes awaken, the cytoskeleton shifts. Immune cells patrol by touch, scanning other cells' membrane-displayed name badges. When a mismatch appears, the membrane becomes a launch pad for defense. Fusion events release toxins. Adhesion molecules grab and hold a target in place. In development and wound healing, adhesion proteins help cells recognize neighbors and align so that sheets become tubes and gaps become whole again. Even the membrane's imperfections are useful. Tiny leaks and noise in channel opening create variability that biological systems harness. A neuron's threshold for firing is not carved in stone. It drifts with chemical context and recent history, making networks flexible learners. In the heart, carefully balanced currents keep the rhythm steady. When channels mutate or drugs interfere, arrhythmias can appear, reminders that a few misplaced ions at a membrane can echo through an entire organ. Scale makes these feats more impressive. The thickness of a plasma membrane is on the order of 5 nanometers. Across that distance, cells maintain voltage differences of tens of millivolts, a gradient comparable to thundercloud fields when scaled to thickness. Proteins within the film sense shifts of a few charges and respond in microseconds. Chemistry becomes circuitry, and circuitry becomes behavior, all in the breadth of a fog's droplet. The story reaches outward too. In tissues, membranes meet at junctions that form sealed belts, preventing leaks between cells and directing nutrients through the proper channels. Other junctions act like doorways, passing ions directly from cell to cell so that a heartbeat can sweep across the heart as a single coordinated wave. Sheets of epithelial cells use their membranes to decide what crosses from the gut into the bloodstream, what stays out, and when to call for help. The integrity of these borders is health itself. What emerges is a simple truth wrapped in beautiful detail. A cell's membrane is a stage for traffic, a ledger for energy, a canvas for signals, and a workshop for remodeling, all while remaining as soft and fluid as a soap bubble. Separate inside from outside, hold a little charge, move with purpose, and cooperate with neighbours. From those steps, life learns to think, to move, and to sense the world. Energy in living things is paid out in small spendable coins called a TP, adenosine triphosphate. Each coin is a compact package, an adenosine head and a tail of 3 phosphates. The last 2 phosphates are crowded and electrically fussy, so when one is clipped off, the reaction releases usable free energy. Cells don't burn ATP like a log, they couple that release to jobs that would not happen on their own, pushing ions uphill across a membrane, bending a protein into a new shape, or stitching molecules together. Enzymes called kinases act like cashiers, transferring the phosphate to other molecules to change their activity. 1,000 tiny purchases each second. Open this channel, close that one, pull here, push there. Every blink, step and thought is paid for in these coins. The scale of the economy is astonishing. Over the course of a day, your cells turn over an amount of ATP roughly equal to your body weight. The same molecules are not hoarded, they are minted, spent and reminted continuously. A single ATP might power a muscle protein for a fraction of a second, become ADP, then be recharged to ATP by the next enzyme along the line. Muscles at rest quietly spend ATP to keep calcium ions sequestered. Muscles in motion spend it far faster as myosin heads ratchet along actin filaments. The brain, though only a small fraction of total mass, is a heavy spender too, using ATP to run the sodium-potassium pumps that reset neurons after each electrical spike. Even when asleep, the budget hums, housekeeping, repair, ion gradients, and the slow work of memory consolidation all have a price tag. Where does the coin come from? Much of it is minted in mitochondria, the powerhouses whose inner membranes host life's most elegant energy trick. Nutrients are broken down to release high-energy electrons, which are passed along a chain of protein complexes. With each handoff, energy is used to pump protons across the inner membrane, stockpiling them on one side and creating a steep gradient, a difference in concentration and charge. That gradient is a kind of stored potential, like water behind a dam. ATP synthase, a rotary enzyme embedded in the membrane, gives the protons a way back, As they flow through, the enzyme turns, mechanically snapping phosphate onto ADP to make fresh ATP. Chemistry becomes machinery, a nanoscopic turbine spinning thousands of times a minute, paid in protons, paid out in energy coins. This system is shared across life. Bacteria run variations on their outer membranes. Plants use sunlight to push electrons and protons in chloroplasts, then pay out ATP to build sugars. In animals, mitochondria sit where demand is highest. In a heart cell, they form a dense network wrapped around the contractile machinery. Each heartbeat requires precise repeated bursts of ATP, myosinate-pases to grab and pull, ion pumps to reset membranes, and calcium pumps, especially the Circa pumps, to clear calcium and relax the muscle for the next beat. Miss a payment, even briefly, and the rhythm falters. Meet the bill, and the heart ticks on, minute after minute, year after year, powered by gradients sustained across membranes only a few nanometers thick. The story of how mitochondria entered the picture is one of biology's best mergers. Long ago, an ancestral cell engulfed a free-living bacterium rather than digesting it. The two struck a partnership. The host provided shelter and raw materials. The bacterium exported ATP with unmatched efficiency. Over time, most of the bacterium's genes moved into the host nucleus, but a few stayed. Mitochondria still carry their own circular DNA, use bacterial-style ribosomes, and divide by fission. They keep a double membrane, a souvenir of their past. The arrangement proved so successful that descendants of that alliance now power animals, plants, and fungi. Inside every cell today is a reminder that collaboration can change what is possible. Mitochondria are not static batteries, they are dynamic. They fuse into elongated networks to share contents, then split apart to isolate damage. When parts wear out, the cell tags the organelle for recycling in a process called mitophagy, disassembly and reuse rather than risky repair. Signals from mitochondria even influence decisions about cell growth and death, tying energy status to fate. In brown fat, a special protein called UCP1 pokes a controlled leak in the proton gradient, letting protons slip back without making ATP. The energy is released as heat instead, a safe, gentle furnace that warms infants and helps mammals adapt to cold. The ATP economy reaches every corner. Tiny motors called kinesin and dynein spend ATP to walk along intracellular rails, towing cargo to where it is needed. Enzymes that copy DNA or repair breaks draw on ATP to clamp and slide along the helix. Cells moving across a surface spend ATP to rearrange their cytoskeleton, pushing the membrane forward and pulling the rear inward. In the immune system, ATP helps power the burst of activity as a cell engulfs a bacterium and digests it. In the senses, photoreceptors in the eye constantly pay ATP to reset after detecting light, and hair cells in the ear use it to maintain the delicately biased positions that make hearing so sensitive. Because the ATP system is so central, small disruptions have wide echoes. A toxin that blocks the electron transport chain starves the gradient and collapses ATP production. A mutation in mitochondrial DNA can lead to muscle weakness or vision problems because those tissues are among the hungriest for energy. Certain drugs must be tested for unintended effects on mitochondria. Even mild interference can reduce stamina or muddle the heart's rhythm. On the other hand, training a muscle increases both the number and efficiency of mitochondria, which is why endurance improves with practice. Cells listen to demand and respond by building more powerhouses or tuning the ones they have. It is tempting to imagine energy as a single grand process, but the body thrives on many linked pathways. Glycolysis in the cytosol can make ATP quickly without oxygen. It is how a sprint begins. The mitochondria take over for the long haul, extracting far more ATP per molecule of fuel when oxygen is abundant. During a fast, the liver conjures glucose from other molecules and releases ketone bodies that mitochondria can burn cleanly in the brain. The flexibility keeps the economy stable through feast and famine, wakefulness and sleep, exercise and rest. And so a basic picture emerges. Energy is stored not in a vault but in a gradient, not in a single place but across a membrane. A rotary machine collects that potential and pays it out as ATP. Enzymes spend the coins one task at a time, and the coins are recharged to be spent again. The system is ancient, borrowed from bacteria and woven into every heartbeat, every breath, every thought. Inside, the lights stay on because countless tiny turbines keep turning, and the currency keeps flowing, coin by coin, quietly funding the business of staying alive. Sunlight is ancient energy arriving fresh each morning, and plants have a way to spend it. Inside their leaves are chloroplasts, and inside those are bright green membranes called thylakoids where photons meet molecules. When light strikes chlorophyll, an electron jumps to a higher energy state and starts a relay race across protein complexes. The most dramatic handoff happens at a place called photosystem 2, where a tiny cluster of manganese atoms and calcium pulls electrons from water. Splitting water is no small feat. It is like borrowing from the toughest bank in chemistry. The price is paid with light, and the reward is oxygen released to the air, plus protons and electrons that the plant can use. Every breath you take carries this signature, oxygen set free by leaves that paid the photochemical bill. Those excited electrons do not simply fade, they move through carriers much like couriers on a narrow path. Plastiquinone shuttles them to a complex called cytochrome B6F, which uses their energy to pump protons into the thylakoid's interior. The effect is a gentle dam of protons on one side of the membrane and a relative scarcity on the other. From there, plastiquinone passes the electrons to photosystem 1, which boosts them with a second photon's push. The high-energy electrons finally reduce NADP plus to NADPH, an electron-rich helper the cell can use for building things. Meanwhile, the proton gradient turns a molecular turbine, ATP synthase, clicking phosphate onto ADP to mint ATP. It is the same turbine design seen in mitochondria, but run by light instead of food. With ATP and NADPH in hand, the plant has currency, energy in reducing power, to turn the faintest gas, carbon dioxide, into sugar. That transformation takes place in the Calvin cycle, a loop of reactions that stitches carbon into stable forms. The key enzyme is Rubisco, a famously slow worker that is nonetheless the world's most abundant protein. It grabs carbon dioxide and tucks it into a sugar backbone, step by step forming molecules that can be stored as starch, burned for energy, or built into cellulose to stiffen stems and trunks. Rubisco has a flaw. It sometimes mistakes oxygen for carbon dioxide, leading to a costly detour called photorespiration. Plants have evolved workarounds. Some species concentrate carbon dioxide near Rubisco by running a clever two-room system known as C4 photosynthesis, separating early steps into outer cells and final steps into inner ones. Others keep their gas exchange largely at night, in a time-separated strategy called CAM, storing carbon as malic acid until daylight. Cacti, agaves, and many succulents use this method to conserve water without starving the Calvin cycle of its raw material. Water management is the daily drama of a leaf. Microscopic pores called stomata open to let carbon dioxide in, but that same opening lets water vapor rush out. Guard cells swell and shrink like gentle valves, responding to light, humidity, internal carbon dioxide, and signals such as the hormone abscisic acid. On a hot, dry afternoon, Many leaves partly close those pores, balancing photosynthesis against hydration. Under cloud cover and cool air, they open wider, drinking in carbon dioxide. Each tiny decision changes the chemistry of the air above forests and fields, and on a planetary scale, those choices tug at the needle of global carbon and water cycles. The green that makes all this possible belongs to chlorophyll, a pigment with a ring-shaped core called a porphyrin that holds a magnesium ion at its centre. That ring system is tuned to absorb red and blue light, reflecting green to our eyes. Swap the magnesium for iron, tweak the surrounding ring, and you have heme, the heart of hemoglobin that carries oxygen through blood. One pigment catches light to free oxygen from water. The other catches oxygen to carry it where it is needed. They are molecular cousins built on the same architectural theme, one tuned for photons, the other for gases. The resemblance hints at evolution's habit of reusing useful designs in new roles. Green is only part of the palette. Carotenoids, with warm oranges and yellows, sit beside chlorophyll in antenna complexes, catching wavelengths chlorophyll misses and protecting it from damage when sunlight comes in hot and heavy. In many algae and cyanobacteria, phycobilins add reds and blues that allow photosynthesis to thrive in waters where light is filtered by depth. Autumn reveals this supporting cast when chlorophyll breaks down. The golds and crimsons and ambers are not new colours but hidden ones, stepping forward as the main actor exits for the season. Look outward, and the quiet arithmetic becomes grand. Every forest, leaf and blade of grass performs the same conversion. Sunlight to sugar, sugar to structure. The sum across a planet is called primary production, and it runs not only on land but in the sea. Floating in the upper ocean are phytoplankton, microscopic plant-like drifters that bloom with sunlight and nutrients. They fix carbon and release oxygen in staggering amounts, shaping climate and feeding food webs from krill to whales. In spring and summer, satellites can watch entire hemispheres inhale as vegetation surges. In fall and winter, the breath reverses. The planet's atmosphere, in part, is a seasonal heartbeat driven by photosynthetic life. Chloroplasts themselves carry a history lesson. They have their own small circular genomes and ribosomes that resemble those of bacteria. They divide inside plant cells the way bacteria divide in open water. These clues tell a story of endosymbiosis. An ancient eukaryotic cell invited a cyanobacterium to stay, and the partnership never ended. Over time, most of the cyanobacterium's genes moved into the host's nucleus for centralized control, but enough remained in the chloroplast to keep it semi-autonomous. The alliance reshaped Earth. Long before animals walked, photosynthetic microbes began releasing oxygen, and over hundreds of millions of years that oxygen climbed, rusting iron out of seas and painting ancient rocks a ruddy red. With oxygen came the chance for efficient respiration, today's abundant ozone shield, and the biological possibilities that followed. Efficiency is modest at the level of a single leaf. Even in good conditions, only a small slice of sunlight is turned into chemical energy, often just a few %. But photosynthesis wins by area and persistence. Leaves thin as paper cover valleys and hillsides, and their work continues day after day, season after season. Plants invest results into wood, roots, seeds, fruits, and the living soil community around them. Fungi and bacteria trade nutrients with roots. In return for sugars, they deliver nitrogen, phosphorus, and trace minerals. In forests, this underground commerce knits individual trees into communities that share resources and information, a web anchored by the steady income of photosynthate. For all its delicacy, the chemistry is robust. A plant can repair damaged photosystems, swap out tired components, and adjust to shade or sun by rearranging its light-harvesting complexes. Shade leaves grow larger, darker and thinner to catch more light. Sun leaves grow sturdier and layered to handle intense illumination without overheating. Alpine plants hug the ground to avoid desiccating winds. Aquatic plants stretch cells to hold gases and drift toward light. Everywhere, the same core mechanism adapts to local demands. The skill of turning light and water into fuel has inspired engineers to pursue artificial photosynthesis, systems that split water, capture carbon dioxide, and make fuels without releasing additional carbon. The dream is a panel or paint that behaves like a leaf, storing sunshine as easily as a tree stores starch. Progress continues, often by borrowing design ideas from chloroplasts, catalysts that do not corrode, membranes that separate charges without shorting, and pigments that harvest a broad spectrum. Meanwhile, the original version works flawlessly outside your window. Light that left the sun about 8 minutes ago lands on a leaf, kicks an electron into motion, and begins a cascade that ends with a sweet molecule and a puff of oxygen. The air in your lungs is a gift from water that was split, atom by atom, by plants and algae. The bite of an apple, the scent of fresh basil, the grain in a loaf of bread, all of it is sunlight rewritten into chemistry, quietly, continuously, leaf by leaf. Beneath a quiet forest floor, a second forest grows, pale, thread-like, and always busy. Roots braid through soil, and from their tips spread filaments of fungi called hyphae, so slender that dozens could pass through the eye of a needle. Where root and fungus meet, they form a partnership older than mammals. The plant trades sugars made from sunlight. The fungus brings back water and hard-to-reach minerals like phosphorus and nitrogen. Some fungi wrap around roots like soft gloves. These are ectomycorrhizae common in pines, oaks, and birches. Others slip inside root cells and build tiny tree-shaped exchange organs. Arbuscular mycorrhizae found across grasses and most crops. Different structures, same bargain. Energy for nutrients paid daily, with interest returned as growth and resilience. Because fungal threads can run far beyond the reach of roots, they stitch together many plants at once. A single fungal individual can link seedlings, saplings, and giants across meters of soil, feeding from one and delivering to another. Through these living cables, resources can move where they are needed most. In cool spring shade, a birch in sunlight may share a little carbon with a neighbouring fir just waking from winter. In deep summer, when the fir's needles feast on full light, the exchange can reverse. Experiments that trace unusual forms of carbon have shown movement from older, established trees to shaded youngsters, a kind of quiet subsidy that helps seedlings survive the dangerous years before their crowns reach the light. Not every plant pair is generous with every other. Partners, species, season, and stress all matter. But the pattern is clear. Roots and fungi together create a community economy. This underground commerce is not only about sugars. Fungal threads explore pores too small for roots, absorbing minerals and passing them back at the trading posts inside the root. In return, plants feed their fungal partners with a steady stream of photosynthate, carbohydrates made from air and light. The cost to the plant can be substantial, a significant slice of its daily income, but the payoff is access to nutrients that would otherwise remain locked in mineral grains or organic debris. In poor soils, the difference between having the right fungal partner and having none can be the difference between a forest and a scrubland. The partnership also changes the soil itself. Hyphae bind particles into crumbs that hold water and air, and fungal glues help stabilize these structures so the ground resists erosion and compaction. Seen this way, a forest is not a set of isolated pillars, but a living web. Every trunk has a shadow network beneath it, and those networks overlap and entwine. Information as well as resources may travel along these routes. When a plant comes under attack, from chewing insects, nibbling deer, or a pathogen at the roots. Its distress can alter the chemistry flowing through the shared fungi. Neighbouring plants connected to the same network often shift their own defences, boosting protective compounds before the threat arrives. It is not telepathy, it is traffic. The same sugars that pay the fungal partner can carry small messages, and the fungus itself can secrete signals that adjust the behaviour of its hosts. Above ground, leaves release volatiles, airborne warnings that drift on breezes, and below ground, the mycorrhizal web seems to play a similar role, but in the slower, denser medium of soil. Chemical warnings can also travel through the air. A leaf bitten by a caterpillar changes its scent almost immediately, releasing a blend of molecules that can make nearby leaves less tasty or more toxic. Some plants call in the enemy of their enemy, scents that attract parasitic wasps, for example, which then lay eggs in the caterpillars. In certain acacia species, trees damaged by browsing herbivores increase tannins in their leaves and release ethylene gas. Downwind neighbours detect the ethylene and prepare their own defences. Willows and poplars send airborne cues that prime neighbours to thicken cell walls or bolster anti-herbivore chemicals. Neither signal is perfect. Wind direction, humidity, and species identity all shape who hears whom, but together, air and soil carry news. The forest is full of eavesdropping. Back underfoot, the fungal network does more than deliver an alert, it also balances water. Hyphae act like wicks, pulling moisture from damp pockets toward thirsty roots. Some deep-rooted trees practice hydraulic redistribution. At night, when transpiration slows, they leak a little water from deep, moist layers into the dry upper soil, where shallow-rooted neighbours can sip it the next day. That redistribution can keep microbes active, which in turn keeps nutrients cycling, which keeps the whole system humming through drought. In this way, a tall tree can behave like a well, slowing the cascade of stress that a hot, dry spell would otherwise bring. The network remembers, not in the way a brain does, but in structure. Paths that carry resources often are strengthened as the fungus invests in those roots. Rarely used paths fade. A successful partner plant grows and feeds more carbon back to the fungus, encouraging more hyphae in its direction. Over seasons, the architecture of the web reflects who is thriving and who is struggling, which species dominate a patch, and how competition and cooperation balance out. When a tree dies, its stored carbon does not vanish in an instant. Some of it flows into fungi and from there to neighbours, some goes into microbes that rebuild soil, some becomes food for insects and animals. Even in decay, the network nourishes the living. Seeds and seedlings gain special advantage from these connections. Many cannot properly establish unless their roots meet the right fungus, and in some forests, seedlings show better survival rates when they germinate near adults of their own species. Part of that success may be shared fungal partners that bridge the gap. In other cases, seedlings of different species benefit from the general mycorrhizal mesh, improving drought tolerance or disease resistance simply by joining the neighbourhood. The phrase nurse tree is not only a romantic image, it describes a real flow of help from those with plenty to those with little, shaped by the shared appetite and reach of fungal threads. Of course, any economy invites tricky behaviour. Some plants attempt to freeload. Orchids, for example, begin life as tiny seeds without enough resources to sprout on their own and rely on fungi to supply carbon until they can photosynthesize. A few adult plants, called mycoheterotrophs, go further and tap the network for a living, drawing carbon indirectly from photosynthetic neighbours via their fungal intermediaries. Fungi have defences, favouring partners that pay well and limiting resources to cheaters. The balance shifts season by season, species by species, but the system endures because most participants benefit most of the time. Because the forest functions as a web, disturbances ripple widely. Remove too many large hosts and the fungal network can thin, reducing nutrient delivery and seedling success. Disrupt the soil with heavy machinery and high-fall highways are torn, slowing exchanges for years. Conversely, enrich the fungal community by leaving fallen logs, avoiding over-tilling at the forest edge, or protecting diverse plant hosts, and the underground fabric thickens. That fabric supports not just trees but ferns, shrubs, mosses, fungi, microbes, and the animals that depend on them. The canopy may look like the main stage, but the orchestra pit is below, and it is always playing. Walk through such a place and the visible story, trunks, branches, leaves, feels complete. Yet the true dynamics hum underfoot, where sugar pays for phosphorus, warnings ride the air and the soil, and water quietly moves in the dark from generosity to need. A forest, seen from this angle, is not a lineup of individuals but a society built on trade routes and shared alarms, with fungi as dispatchers and translators. The next time wind moves through leaves, remember that a conversation is flowing below as well. An exchange measured not in words, but in molecules, shaping who grows, who survives, and how the whole stays alive. Plants move without muscles, and once you learn the cues, you start to see it everywhere. A young sunflower spends the day turning its face from east to west, leaning into the light as if listening. The trick is unequal growth. Light-sensitive proteins in the stem read the direction of the brightest rays, and a hormone called auxin shifts toward the shaded side. Cells there elongate slightly more than those on the lit side, and the stem bends toward the sun. By evening, the head faces west. Overnight, a built-in clock rewinds the posture so the plant greets dawn from the east again. This rhythm fades as the flower matures and the heavy seed head settles into a steady eastward pose. Warmer mornings bring more pollinators. Vines use a different tactic. They sweep slow circles, a motion called circumnutation, until touch tells them they have found an anchor. Specialized cells sense the mechanical nudge. Growth slows on the touched side and speeds on the far side, and the tendril curls. In a few hours, a gentle coil becomes a spring that can lift the plant toward better light. Many plant motions are not growth at all, but hydraulics, moving water to move tissue. At the base of certain leaves and leaflets sits a tiny joint called a pulvinus, packed with cells that can rapidly change their internal pressure. When potassium and chloride ions flow out, water follows, pressure drops, and the tissue slackens. When ions flow back in, water returns and the joint stiffens. This is how some leaves open by day and fold by night, a behavior called nitinasty. It is also how they respond to touch or wind. The sensitive plant, Mimosa pudica, collapses its leaflets within seconds of a tap because stretch-activated channels trigger an electrical signal that sweeps through the pulvinus, shifting ions and with them, water. No muscle fibers are needed, just valves, pipes, and a little electricity riding a living membrane. At the faster end of the spectrum sits a botanical celebrity. The Venus flytrap looks like a green clam with eyelashes until something brushes those hairs. 2 touches within a short window confirm that the stimulus is likely prey and not just raindrops. That double tap launches an electrical pulse and a mechanical trick. The trap's 2 lobes are primed like a bent metal snap bracelet. When the pulse changes cell pressure along the mid-rib, the geometry flips from convex to concave in a blink, and the cilia-like teeth interlock. If the struggle continues, more hairs touched, more signals fired, the plant seals the edges, pumps fluid into the trap to tighten the fit, and releases digestive enzymes. Days later, the trap reopens, leaving a clean husk. Venus flytraps live in nutrient-poor bogs. By hunting, they supplement scarce nitrogen and phosphorus. Each trap is precious hardware and can only close fully a limited number of times before it wears out. So the double touch rule avoids wasting a meal on a false alarm. The fly trap is not alone. Sundews curl sticky tentacles around trapped insects, moving faster where a signal has been triggered and slower where it has not, a gradient of motion guided by chemistry. Bladderworts set hair-trigger doors on underwater bladders. Brushing the hairs opens the door and the bladder's vacuum sucks in a rush of water and snacks. In the deserts of the Americas, the so-called telegraph plant twitches tiny leaflets in regular arcs, a slow semaphore powered by ion flows and turgor changes. Each is a different instrument playing the same tune. Convert a small environmental cue into an electrical signal. Convert that signal into an ion shift and let water and cell walls do the rest. Even when nothing dramatic happens, plants are busy. Stomata, those microscopic pores on leaves, open and close by adjusting the pressure in paired guard cells, balancing the need for carbon dioxide against the risk of losing too much water. Chloroplasts inside leaf cells reposition themselves through the day, gathering near the cell surface under dim light to catch more photons, retreating along the walls under harsh light to avoid damage. Roots steer around obstacles and toward nutrients by sensing gradients of moisture, oxygen, and dissolved minerals. They thicken or thin, branch or hold steady, according to the story the soil tells. Pollen tubes grow as single cells that can navigate down a stile, turn corners, and respond to chemical signposts from the ovule, homing with uncanny precision. Cytoplasmic streaming, a slow river driven by motor proteins on internal filaments, ferries, organelles around large cells so resources do not sit stranded. Touch is a language plants read fluently. A climbing bean knows the difference between a rough stake and a smooth wall. The degree of friction and the shape of the support change how quickly it coils and how tight the spiral grows. A carnivorous pitcher plant senses rain, wind, and visiting insects differently. Its slick inner surface becomes more or less slippery depending on humidity, a passive but effective shift. Even sound and vibration can matter. Pea roots have been shown to grow toward the faint splash of water moving through soil, and some flowers increase nectar sweetness after the buzz of a bee's wings. Mechanical cues folded into the plant's decision-making. Speed in plants often comes from structural tricks. Some seed pods dry into shapes that store tension. A touch or a rise in heat is enough to spring them open, flinging seeds meters away. The fern sporangium dries from the outside in, a ring of cells acting as a natural catapult that hurls spores into the air when conditions favour dispersal. These movements are not reversible like a muscle flex, but they are repeatable across the population and exquisitely tuned to humidity and temperature. The engineering is elegant. Built-in hinges, buckling panels, and layered tissues that expand at different rates. The slow pace of many plant motions hides their complexity, but time-lapse reveals a choreography as rich as anything in animal behaviour. A sunflower field does not simply stand, it breathes in light, tracks it, and settles into optimal positions as the season turns. In a hedgerow, brambles seek footholds, testing and releasing, testing and clinging. In a meadow, clover leaves tilt and rest in an easy rhythm tied to the day-night cycle. The unifying power source is turgor pressure, the gentle push of water against cell walls, coupled to clever plumbing and the sensitivity of ion channels. Where an animal might deploy fibers that slide, a plant deploys compartments that swell and slacken. Because plants lack nerves in the animal sense, their electrical signals were once overlooked. They are real and measurable. A wound can send a wave of voltage through a leaf and into the stem, prompting distant tissues to boost defences. A touch can excite a series of action potentials that travel along a vein network, coordinating a compound response across a leaf's many sections. The speeds are slower than a mammal's nerves, but the logic is recognizable: detect, transmit, respond. The result of all this, growth steering, pressure shifts, snap mechanisms, and long-distance signals, is motion that suits a rooted life. Plants do not walk to water, so they redirect their roots. They cannot chase the sun, so they shift stems and leaves to meet it. They cannot run from herbivores, so they close, curl, toughen, or call for allies. It is a different toolkit aimed at the same goal any organism pursues: to find resources, avoid harm, and make the most of the day. Watch a patch of garden across a week, and you will see it. Tiny decisions adding up to graceful change. Movement is not the exception in plants, it is the rule, written in water and made visible by patience. A seed is a pause button pressed at exactly the right moment. Inside a shell hardly larger than a fingernail sits an embryo, a packed lunch of starch and oil, and a kit of molecular tools for waking up when the world says now. Some seeds take later very seriously. In rare cases, dormancy has lasted for thousands of years. Dryness, cold, and darkness act like 3 locks on a door, until the keys arrive: rain, warmth, light. The door stays shut and the little plant waits. Waiting is an art. As moisture slips away during maturation, cells replace liquid water with glassy sugars that immobilize proteins and protect membranes. Specialized proteins, often called late embryogenesis abundant proteins, brace the cell's machinery like bubble wrap. Metabolism slows to a creep. Free radicals that would normally chew at DNA are held at bay, and DNA repair enzymes stand ready for the moment a first sip of water arrives. When it does, the embryo's chemistry rehydrates and unfreezes. Enzymes flicker back to life, mitochondria reboot, and the seed begins to breathe again. Dormancy is not only about preserving the seed, it is about choosing the correct calendar. Plants lace their seeds with chemical stop signs, abscisic acid to prevent premature sprouting, for instance, and conditions must erode those signs before germination proceeds. Cold stratification in winter, or cycles of wet and dry, leach inhibitors and rewrite the hormonal balance. Some species demand abrasion, a coat so tough it must be sanded by river gravel, or scraped in the gut of a passing animal before water can enter. Others listen for smoke. After a wildfire, airborne molecules called charikins settle into soil and tell buried seed banks that competition has been cleared, nutrients are ash fresh, and light will reach the ground. The same hillside that seems silent becomes a pageant of sprouts because the seeds kept their patience. Not all dormancy is long, some is strategic. In deserts, a plant may produce seeds with mixed settings, some eager, some cautious. A small fraction germinates after the first good rain. The rest hold back for the next storm, or the one after that, spreading risk across seasons. This bet hedging keeps the species alive when rainfall is unpredictable. In temperate forests, seeds may insist on a winter chill before they will wake, ensuring that a warm spell in autumn does not trick them into emerging only to be killed by frost. Timing is everything when you are rooted to the spot you pick at birth. At the other end of the clock, speed arrives the instant dormancy lifts. Bamboo is famous for overnight transformations, a reminder that most growth is not cell division, but cell expansion. The plant has already built the blueprint and many of the cell walls in the culm while it was still sheathed and short. When the rains and warmth align, the plant pumps water and solutes into those preformed cells, the walls loosen, and the tissue inflates like thousands of tiny accordions opening at once. Internodes lengthen, sometimes nearly a meter in a day for the fastest giants, powered by hydraulic pressure and fueled by sugars shipped in from the underground rhizome network. Those rhizomes are the quiet accountants of the bamboo world, spreading laterally, storing carbohydrates, and choosing where to invest. A shoot thrust up into the light is a calculated spend from savings. The engineering is clever. Microfibrils of cellulose in the cell wall are angled so that when pressure rises inside, the wall stretches lengthwise more than it widens, producing height rather than floppy girth. Enzymes called expansins slip between wall fibers and reduce the friction that resists stretching. As the growth spurt finishes, lignin hardens the tissues, locking in the new form. The result is a plant that can overtake a window in the time it takes to binge a television series, not by frantic building, but by intelligent unfolding. Bamboo has another dramatic clock, the life cycle of flowering. Many species are synchronous and simulpareous. Whole hillsides may flower at once after decades of vegetative quiet, set seed in a rush, and then die back together, leaving a legacy carpet for the next generation. The rhythm is so steady that historical records have tied famine and rodent booms to these rare massed events. The logic is brutal and brilliant. Overwhelm seed eaters with more food than they can handle in a single season, ensuring that many seeds escape and sprout. The stand sacrifices itself so its children can own the clearing. Step back to the seed again, and you start to see why compact memory of sunlight and soil is more than a poetic line. The embryo carries anatomical memory, a root tip with a cap that will sense gravity, a shoot apex that will read light. The storage tissues reflect the plant's ecological plan, oily in some species for high-energy starts, starchy in others for a steady burn. The coat's chemistry encodes the likely challenges: tannins against fungi, waxes against water loss, pigments that block ultraviolet. Even the seed's size is a bet. Large seeds land with enough supplies to punch through leaf litter and shade. Small seeds travel far and can sift into tiny cracks that big ones miss. There's also a memory of the mother plant's life. Conditions during seed development, day length, temperature, soil nutrients, leave hormonal and epigenetic marks that tune dormancy and early growth. A dry season can produce offspring primed to conserve water. A cool summer can bias seeds toward waiting for a longer winter before they risk germination. These tweaks do not rewrite the DNA sequence, but they adjust which genes turn on readily and which stay quiet, nudging the next generation toward better odds. And then there is the memory of movement. Seeds are designed to leave. Maple keys helicopter on a breeze. Dandelion parachutes ride thermals to the next lawn. Burrs hitchhike on fur and socks with microscopic hooks inspired enough to invent Velcro. Fleshy fruits bribe birds and mammals with sugars and colour. Seeds pass through the digestive tract scarified and fertilised, deposited with a bit of manure in a sunny patch. Some species launch themselves. Squirting cucumber fruits build pressure and fire their contents in a wet burst. Touch-me-not pods coil and fling seeds when bumped. Each strategy writes a different map across the landscape. The soil itself keeps records in a living archive called the seed bank. Under your shoes lie thousands of unsprouted possibilities from seasons past, some viable for a year, some for a decade, a few for far longer. Disturb the ground, let in light, change the moisture, and new species seem to appear out of nowhere. They were always there, filing cabinets of time stacked just below the surface. Land managers know this well. Restoration can coax back native plants by adjusting conditions, while careless disturbance can invite waves of opportunists long waiting for their chance. Germination, when it comes, is not a single act but a sequence. Water soaks in and swells the tissues. The coat cracks along pre-designed seams. The first rootlet, the radicle, pushes out and anchors, guided downward by statoliths, tiny starch grains that settle with gravity and tell cells which side is down. The hypocotyle or coleoptile rises, pulling the cotyledons with it, which in some species are true leaves and in others are specialized storage organs. Enzymes wake and begin to liquefy starch into sugars and protein into amino acids, feeding rapid growth until the first real leaves can catch light and the plant transitions from spending its inheritance to earning its income. From there, forests, fields and gardens are simply scaled-up unfoldings of that same plan, the acorn cracking into a sapling, the wildflower carpeting a verge after the snow recedes, the cereal grain rising into a crop that feeds a city. Each began as a hard, small promise. Seeds knit seasons together. They allow a plant to endure cold, drought, fire, and time itself by stepping off stage and then returning on cue. In their patience and their sudden vigour lies a kind of resilience that animals rarely need and cannot match. The next time a seed sticks to a sleeve or a breeze lifts a tiny parachute past your window, consider what rides inside. An embryo with a toolkit, a pantry, a weather forecast, and a willingness to wait as long as it takes. Inside the skull sits a forest of activity. Roughly 86 billion neurons, each shaped for conversation. Some are compact with starry branches, others stretch astonishing distances, a single cell carrying a signal from the spine to the toes. Their language is electrical. A tiny change in voltage opens channels in the membrane, and an impulse travels along the axon in a wave that lasts only thousandths of a second. At the end, the signal becomes chemical for the briefest moment. Neurotransmitters cross a synapse to the next cell, then turns electrical again. Trillions of these junctions form living circuits that can shift and strengthen with experience. Learn a song, meet a friend, recall a smell from childhood, and certain pathways become a little easier to travel the next time. The brain is not a filing cabinet. It is a city that brightens in different neighbourhoods depending on what you ask of it. Thoughts and feelings are not things floating in the air. They are patterns moving across tissue. Happiness is a choreography in networks that process reward and attention. Fear is a fast route that prioritises speed over detail. Focus is a quietly humming loop between deep structures and the frontal cortex. Memory is the art of changing connections. Long-term potentiation nudges certain synapses to pass signals more readily, so a once faint path becomes a well-trodden one. Even at rest, the brain is busy, wandering through a default mode that knits together self, time, and story. Glial cells, often the unsung partners, feed neurons, mop up stray chemicals, insulate axons with myelin to speed signals, and prune connections that no longer serve. The whole network runs on a constant energy stipend, burning a surprising share of daily fuel simply to hold itself at the edge of readiness, poised for the next idea. Far from the head, another metronome works with its own beat. Heart muscle contains pacemaker cells clustered in the sinoatrial node. They leak ions in a steady rhythm, depolarize, and trigger contraction without any need for orders. That intrinsic clock is why a heart, freshly supplied with oxygen and warmth, can beat outside the body for a short time. The timing is built into the tissue itself. The signal spreads through specialized pathways, reaching the atrioventricular node, then racing along branching fibers so the ventricles squeeze from the bottom up, a neat trick that maximizes each pump. Autonomic nerves and hormones tune the speed, accelerating for a sprint, slowing for sleep, but the fundamental pulse begins within. In a single minute, this simple, relentless pattern pushes blood through vessels that, if laid end to end, would span continents and back, delivering oxygen and returning carbon dioxide with machine-like reliability and living grace. The gut plays its own music. Smooth muscle rings contract in waves called peristalsis, moving a meal along a carefully timed conveyor. The stomach's lining secretes acid and enzymes powerful enough to break down protein. Yet the tissue survives by renewing itself rapidly. Cells near the base of gastric glands divide and migrate upward, replacing the surface every few days so the wall does not digest itself. Farther along, in the small intestine, absorptive cells covering finger-like villi also turn over swiftly. New cells climb from crypts, do their work, and are shed into the lumen, where they are recycled. The enteric nervous system, sometimes called a second brain, coordinates many of these motions and secretions locally, taking broad suggestions from the brain but improvising the details based on what arrives for digestion. Bone seems solid and done, but it too is alive with change. Under the microscope it is a matrix shot through with blood vessels and threaded with cells. Osteoclasts dissolve old or micro-damaged bone. Osteoblasts lay down fresh mineral and collagen in its place. The process is responsive. A tennis player thickens the serving arm's humerus. An astronaut without the tug of gravity loses mineral until weight-bearing resumes. Tiny electrical signals generated when bone flexes help guide the remodeling crew to where reinforcement is needed. Over months and years, the skeleton quietly edits itself to match the life it carries. Skin, our largest organ, provides a barrier that is always in motion. New keratinocytes form in the basal layer and travel outward, filling with the tough protein keratin as they go, until they flatten into the stratum corneum and eventually slough away. Pigment-making melanocytes adapt the tone in response to sunlight, and immune cells patrol the layers to catch intruders trying to slip through. A scrape triggers a fast response. Platelets form a clot. Nearby cells release signals that call in repair teams. Edges contract as fibroblasts lay collagen. New epidermis creeps over the gap. The surface looks calm, but the workforce never clocks out. Across all of this, the theme is renewal balanced by memory. Neuronal pathways keep the essence of yesterday's learning, while remaining flexible enough to absorb today's. The heart maintains a rhythm so dependable that slight irregularities can feel like thunder. The stomach and intestines rebuild quickly because their environment is harsh. Bones remodel slowly because their job is load-bearing. Skin strikes a middle pace, changing fast enough to defend but slow enough to hold form. Specialized systems handle emergencies. A sprint borrows fast ATP from stored molecules, then pays the debt with deeper breathing. A fever cues the liver to change its chemistry. A cut launches clotting and cleanup, then signals the danger has passed so scar does not overgrow. It is tempting to imagine a command center issuing orders, but much of the harmony emerges from local rules. Cells open channels when the voltage crosses a threshold, secrete enzymes when stretch receptors report fullness, divide when neighbor signals say there is room, halt when contact is made. From these simple decisions, whole-body behavior arises. Focus narrows because certain neuronal assemblies are encouraged, while others are damped. Calm spreads because the vagus nerve shifts the balance toward rest and digest. Wakefulness fades as adenosine builds, and receptors nudge networks towards slower rhythms. Even sleep fits the orchestra. While consciousness dims, cerebrospinal fluid pulses through the brain more freely, washing away byproducts of a busy day. Synapses that were briefly strengthened during learning are trimmed and consolidated, making room for tomorrow's changes. The heart adopts a slower tempo. The gut takes advantage of the lull to move along housekeeping waves. Skin repair accelerates. Nothing truly stops. Everything changes its tempo to suit the hour. So the body's music is not a single melody but a layered score. Electrical whispers among billions of neurons create the shifting landscape of mind. A self-starting drum in the chest drives blood, moment after moment, without rehearsal. Tissues wear and renew, some in days, some across seasons, their rhythms tuned to the demands they face. The sensation of being one person comes from this quiet unity. Many instruments, one piece playing on. Most of life is too small to notice, and yet it runs the show. Bacteria, archaea, single-celled eukaryotes, and viruses fill every niche that water can touch, and many that it barely can. They thrive in boiling springs and under glaciers, in clouds and caves, in the thin films on leaves and the films inside your mouth. Take a handful of lake water or a breath of sea spray and you are holding worlds, communities that photosynthesize, hunt, graze and recycle in a dance that has been continuous since long before animals appeared. The great tree of life is a bush whose longest branches are microbial. Animals and plants are recent twigs at the edge. Soil makes the point in a single teaspoon. Under a leaf, in a garden bed, inside a forest floor, scoop and you will find more microbes than there are humans on Earth. Not just more individuals, but staggering variety. Some specialize in nibbling bits of lignin from dead wood. Others crack open the rings of aromatic pollutants. Others trap nitrogen from the air and trade it to roots for sugar. Fungi thread through the mix as long white filaments. Bacteria build cities called biofilms on grains of sand, and archaea carry out chemistry so odd that it took decades to realize they are neither bacteria nor anything like us. This underground crowd is why a fallen branch becomes soil, why a meadow can bloom again after winter, and why a compost pile warms its own hands. Microbes are not merely decomposers, many are sunlight's first accountants. In the ocean's sunlit skin, drifting microbes called phytoplankton fix carbon and release oxygen, launching food webs that climb to fish, seabirds, and whales. Far below in darkness, others eat minerals instead of light. They oxidize iron, taste sulfur, and live at the mouths of hydrothermal vents, where chemical energy gushes from the Earth's crust. Life began without eyes on such transactions. It still leans on them. The scale turns personal when you consider the company you keep. Your body is an apartment block of habitats: skin, mouth, lungs, gut, and each is home to its own communities of microbes. Together they amount to trillions of cells, rivaling your own cells in number and easily surpassing them in genetic diversity. On skin, residents vary with climate and landscape. Dry forearm deserts, oily forehead plains, humid armpit forests. In the mouth, ecosystems cling to the enamel and the tongue's papillae. But the largest, busiest metropolis runs through the intestine, where a dense, oxygen-poor environment supports fermenters of every persuasion. Digestion is the most obvious benefit. Microbes break down complex fibers that human enzymes cannot touch, turning them into short-chain fatty acids such as acetate, propionate and butyrate. Those small molecules feed the cells lining the colon, strengthen the barrier that keeps inflammatory compounds from leaking into the blood, and nudge the immune system toward calm vigilance rather than chronic alarm. Symbionts synthesize vitamins, including K and several B vitamins, top up amino acids, and metabolize plant chemicals into forms we can absorb. When diets change, the cast of characters shifts. A menu rich in diverse plants tends to encourage a more diverse, resilient community, while one-note diets can shrink the repertoire. Immunity is shaped here too. Early in life, encounters with microbes teach immune cells the difference between friend, foe, and harmless passerby. Specialized cells in the gut's lymphoid tissue sample the flow and present pieces to T cells, like a careful teacher introducing a shy class to the neighborhood. Signals from microbes help set the balance between attack and tolerance. Without these lessons, the system can skew toward overreaction, think allergies, or misdirect its fire at the self. Even later, the conversation continues. Molecules from bacteria, including those short-chain fatty acids, guide regulatory T cells that keep inflammation in check. Mood may seem far away from the colon, but the root's direct. Microbes influence the production of serotonin by gut cells, tweak the sensitivity of vagus nerve pathways that carry signals to the brain, and manufacture small metabolites that slip into the bloodstream and cross into neural territory. They do not author thoughts, but they colour the background. People notice the effect when an illness, antibiotic course, or dramatic dietary change leaves them vaguely off. Sleep unsettled, appetite wrong-footed, patience thin. Put the community back in balance, and the weather inside stabilizes. The microbiome is not static, it is a weather system. It changes with age, travel, stress, pets, exercise, seasons, and the antibiotics we take. Some antibiotics are life-saving. They also mow down bystanders. After such a sweep, hardy opportunists may surge until the original diversity grows back. This is one reason clinicians are cautious with broad-spectrum drugs, and sometimes pair them with very specific ones or with probiotic strategies. Fermented foods, yogurt, kefir, sauerkraut, kimchi, do not permanently stock you with new citizens, but they can serve as friendly visitors that nudge the locals, supply metabolites, and occupy seats that might otherwise be claimed by less helpful guests. Fibre, meanwhile, is food for the microbiome. What you do not digest, they do, and their byproducts feed you in return. Microbial life has a talent for swapping know-how. Bacteria can trade genes sideways in addition to passing them down, handing around little DNA circles called plasmids that carry tricks like antibiotic resistance or the ability to digest a new sugar. In a hospital or a farmyard, this flexibility is part of why resistance evolves quickly. A few cells survive a drug, share their advantage, and the next generation is tougher. It is also why microbes adapt to new pollutants and break them down faster than expected. The right enzyme can spread through a population like good gossip. They are engineers as well. In wetlands, communities knit together mats that trap sediment and create new layers of land. In the human mouth, a dental plaque is not just grime but a biofilm with architecture, channels for nutrients, sheltered neighbourhoods, cooperative chemistry that makes the group harder to wash away than any single cell would be. On medical implants, similar films can be stubbornly persistent. The same stick-together-to-survive strategy that makes microbes great at living everywhere also makes them, at times, formidable to treat. Numbers matter, but so do neighbourhoods. In healthy people, many potentially harmful species live peacefully at low levels, held in check by competitors and by the immune system. Trouble tends to arise when balance is disturbed, after antibiotics, during severe stress, with drastic dietary swings, or when the immune system is compromised. Then a usually minor player can bloom, like an invasive plant after a fire. Restoration is about rebalancing, reintroducing diversity, feeding the helpers, and letting checks and balances resume. Step back, and the portrait of life tilts. The creatures most responsible for cycling carbon and nitrogen, for keeping soils fertile, for much of the oxygen in the air, for the vitamins in your blood and the calm in your gut. Most of them are invisible. They are not an other living on us. They are part of the us that functions. Wash your hands when you should, cook your food properly, be wise with medicines, and then, mostly, be a good host. A diverse, well-fed community of tiny partners keeps the larger one, the human, running smoothly. The world at microbial scale is busy, creative, and generous, and it touches every breath and every meal, whether we notice or not. Partnerships built the modern cell. Long before animals and trees, an ancestral cell struck a deal with a free-living bacterium that was spectacular at turning food into energy. Instead of digesting the visitor, the host kept it, fed it, and routed chemical traffic around it. The bacterium paid rent in ATP. the tiny energy coins that keep metabolism humming. Over time, most of the bacterium's genes moved into the host's nucleus for centralized management, but handfuls stayed behind. What remains today is the mitochondrion, still dividing on its own schedule, still carrying a small circular genome, still wrapped in a double membrane that hints at the ancient gulp that brought it inside. When you read that mitochondria resemble bacteria in their ribosomes and split by fission, you are hearing the forensic details of a merger written into the hardware of every animal, plant and fungus cell. Plants carry a second symbiotic signature. Their chloroplasts, the green factories that convert light into sugar, also trace back to bacteria, this time a cyanobacterium capable of photosynthesis. The story likely unfolded in 2 acts. First, the acquisition of mitochondria, which turbocharged energy metabolism, and later, the adoption of a photosynthetic partner, which opened an entirely new economy powered by sunlight. Some algae show a twist called secondary endosymbiosis, where a non-photosynthetic host swallowed an already photosynthetic eukaryote, layering membranes like a set of nested dolls. If mitochondria and chloroplasts seem indispensable now, that is because cooperation didn't just help life, it reinvented what life could be. From there, the pattern repeats at every scale. Licens, which crust rocks in polar deserts and paint tree bark in temperate forests, are not single organisms but living coalitions, a fungus for structure and shelter, an alga or a cyanobacterium for sugars, sometimes extra microbes for vitamins and odd jobs. Together they colonize places too harsh for either partner alone, dissolving stone grain by grain and creating the first flecks of soil. Coral reefs are likewise built on a handshake. Reef-building corals house photosynthetic dinoflagellates within their tissues. By day, the tiny tenants pay rent in sugars. By night, the coral animals cast out stinging tentacles to capture passing plankton, earning protein to complement the daytime carbohydrate income. The resulting economy is so productive that reefs, though they cover a tiny fraction of the ocean floor, feed, shelter, or nurse a vast share of marine life. Symbiosis runs through the animal world in quieter forms too. Ruminants like cows digest tough plant fibers with the help of microbes that ferment cellulose into fatty acids the host can absorb. Termites rely on protists and bacteria in their gut to tackle wood. Leafcutter ants grow fungus gardens underground, cutting fresh leaves not to eat directly but to feed their crop. The ants harvest the swollen fungal tips as food and carry antibiotic-producing bacteria on their bodies to suppress molds that might invade the garden. A Hawaiian bobtail squid hosts a single species of bioluminescent bacteria in a specialized light organ. The glow erases the squid silhouette at night, hiding it from predators below. Each partner trades something the other struggles to produce: enzymes, shelter, nutrients, or defense, and each becomes more than it could be alone. Plants make similar deals. We have already visited the world of mycorrhizae, fungi exchanging minerals for sugars with roots, but the partnerships extend further. Some plants host nitrogen-fixing bacteria in nodules on their roots, paying them with carbohydrates in return for ammonia that the plant can turn into proteins. Without these alliances, many soils would be poor. With them, entire ecosystems flourish. Even single cells can collaborate. In the ocean, some diatoms and nitrogen-fixing cyanobacteria live cheek by jowl, trading carbon and nitrogen in a microscopic market that feeds larger food webs. Partnerships can become so tight that the lines blur. Aphids harbour tiny bacteria called Buchnera that supply essential amino acids missing from their sap diet. Over millions of years, the bacteria have shed genes until they can no longer live independently. The aphid, in turn, has evolved to house and transmit them. Deep-sea tube worms at hydrothermal vents have no mouth or gut at all. Bacteria inside their tissues oxidize sulfur compounds and hand over the profits. The worms offer safe housing and chemical substrates, and in exchange they gain a complete metabolism. What starts as cooperation can, given time, become identity. Symbiosis explains strange hybrids too. A small sea slug famously steals chloroplasts from the algae it eats and keeps them functioning in its own cells for weeks, turning sunlight into a snack between meals. Some flatworms host algae within their tissues and bask in the daylight to harvest sugar like tiny living solar panels. In each case, boundaries soften just enough to borrow talent without surrendering autonomy entirely. Cooperation over competition does not mean nature is gentle. It means that success often depends on managing conflicts inside partnerships. Cheaters appear, bacteria that cache the host's benefits without paying, plants that tap fungal networks heavily while returning little sugar, reef symbionts that overgrow when the balance tips. Successful alliances evolve policing and feedback. Corals expel symbionts that misbehave or simply fail under heat stress, which is what bleaching really is, a forced divorce when conditions break trust. Legume plants limit oxygen in root nodules to favour nitrogen fixation and can throttle resources to nodules that underperform, rewarding the most generous microbial strains. In leafcutter gardens, ants weed and medicate, actively engineering their ecosystem to keep it cooperative. Even your own immune system acts as a peacekeeper, rewarding helpful neighbours with tolerance and flagging threats for removal. The microbiome as a whole functions because the host supplies mucus, secretions and surfaces that favour certain communities. The communities in turn provide vitamins and anti-inflammatory metabolites. If a member oversteps, antimicrobial peptides and competitive neighbours nudge it back, restoring balance. Stability arises not from the absence of conflict but from negotiated truce. Cooperation also accelerates evolution's creativity. Endosymbiosis is gene transfer writ large, but microbes move genes laterally all the time, sharing enzymes that break down new sugars or resist antibiotics. In coral reefs, the algal partners can shift over generations toward more heat-tolerant strains, potentially buying the host time in warming seas. In insects, symbionts can confer resistance to parasites or heat, traits that spread quickly because they ride from mother to offspring within eggs. Evolution is not only mutations filtered by survival, it is also mergers, loans, and communal innovation. Look closely and the moral is simple. Life's big leaps, the eukaryotic cell, photosynthetic plants, hyperproductive reefs, arrived when separate lives stitched themselves together. The proof is in every breath and every meal, mitochondria paying out ATP for movement and thought, chloroplasts laying down sugar from light, microbial partners making soil fertile, algae feeding a city of coral animals, gut bacteria finishing the chemistry our enzymes begin. Cooperation is not a footnote to natural selection. It is one of its preferred tools. When living things combine strengths and share the spoils fairly, new worlds appear, sometimes inside a cell, sometimes across an ocean, always with consequences far larger than the partners imagined. Life does not flinch at the edges we draw on maps. In near-boiling pools where the water fizzles with acid, microbes, bacteria and their cousins, the archaea, go about their business as if the spa were set too pleasant. Their proteins are braced with extra bonds, their membranes are built from sturdier lipids, and their enzymes work best at temperatures that would turn an egg solid in seconds. In other corners of the planet, cold is the challenge. Psychrophiles live in the brine channels of sea ice, where salt lowers the freezing point and thin films of liquid thread through a frozen world. Their enzymes remain flexible at low temperatures, and their cell membranes stay fluid by packing in unsaturated fatty acids. the biological equivalent of adding antifreeze to a car. Move from cold to crushing pressure, and another guild appears, piezophiles that thrive far below the surface, in trenches where the weight of the ocean stacks like mountains of air above them. Their proteins are shaped to resist being squeezed. Their membranes are tuned so that motion remains possible in a world of force. Radiation, which shreds DNA and unravels molecules, looks like a final boss, until it doesn't. Certain bacteria withstand doses that would make human cells give up instantly. They do not block every ray, they endure and repair. Their DNA is arranged in tight rings and often present in multiple copies. When damage scatters fragments, molecular repair crews fetch matching pieces from the spare copies and stitch the genome back together with astonishing fidelity. Pigments and antioxidants mop up reactive byproducts before they can do harm. Even in places humans label radioactive, such as reactor cooling pools and waste storage sites, microbial films grow, graze and recycle, turning a hazard into a habitat. Elsewhere, in caves black as pitch and older than written history, microbes take electrons from minerals and breathe what is available. Food and air become flexible categories when chemistry is on your side. Some of the most surprising survivors look like plush toys. Tardigrades, water bears, are small eight-legged animals that trundle through moss and puddles on chubby feet. Under stress, they perform a trick that seems like fiction until you watch it happen. They shut down and wait. The animal curls into a ton, a desiccated capsule that looks like a deflated football. Inside, metabolism drops to a whisper. Proteins that would normally denature without water are stabilized by special sugar alcohols and a set of unique, glass-forming proteins that act like molecular packing peanuts. Membranes are buttressed, DNA is protected, and reactive molecules are neutralized. In this state, tardigrades have endured vacuum, survived bursts of radiation, tolerated deep cold and intense heat, and even returned from exposure to space, harsh sunlight, emptiness and all, when rehydrated back on Earth. The point is not that they enjoy these conditions, it is that they can wait them out and then rejoin the ordinary business of life when the weather improves. The theme running through all of these feats is adaptation at the level of molecules. Proteins must flex to work, but flex too much and they fall apart, flex too little, and chemistry stalls. Thermophiles solve the heat problem by adding stabilizing interactions and salt bridges that keep enzymes tight at high temperatures. Psychrophiles solve the cold problem by loosening the same structures so that reactions can proceed in the slow dance of the chill. Halophiles that live in salt flats where crystals grow at noon make their insides salty too, balancing the outside world rather than fighting it. Acid lovers keep their cytoplasm near neutral pH, while porting protons in and out as needed. Alkali lovers do the inverse. Every extreme looks like an impossible barrier until you realize that life does not bring a fixed toolkit to the environment. It edits the toolkit to match what the environment demands. Membranes tell a similar story. The oily layer that encloses a cell has to remain intact across pressure, heat, cold, and salt swings. Archaea swap the ordinary building blocks for ether-linked lipids, chemically tougher than the ester-linked lipids in many bacteria and eukaryotes. Some even fuse their lipids tail to tail to make monolayers instead of bilayers, creating a single continuous sheet that will not peel apart in heat. Under pressure, deep-sea microbes increase unsaturated and polyunsaturated lipids to keep the membrane from solidifying. In concentrated brines, certain microbes pack compatible solutes, small inert molecules, inside to balance the osmotic pull without gumming up the works. The membrane remains a conversation surface and a power storehouse, even when circumstances try to turn it into a rigid wall or a sieve. Metabolism, the basic traffic of electrons and atoms, also stretches across extremes. Where sunlight is scarce, chemolethotrophs eat rocks in the most literal sense. They strip electrons from iron, sulfur or hydrogen, then hand those electrons to acceptors like oxygen, nitrate, or even metals, harvesting energy in the process. In the deep sea, entire communities gather around hydrothermal vents, where volcanic chemistry feeds microbes that in turn feed giant clams, white crabs, and red-plumed tube worms. In darkness under Antarctic ice, bacteria graze on oxidants formed when cosmic rays and trapped salts work slow magic. The planet supplies gradients of heat, of chemicals, of acidity, and microbes find ways to ride them. All of this expands the map of where life might be found beyond Earth. If cells can keep going in the briny veins of sea ice, maybe similar brines on Mars once hosted chemistry that learned to copy itself. If microbes can live at crushing pressures around hydrothermal vents, icy moons with inner oceans, worlds like Europa and Enceladus, look less like barren balls and more like plausible habitats. If desiccation can be endured for years by entering a glassy protected state, then spores or tons might ride dust and survive long, cold transfers between niches. Speculation is not proof, but extremophils move the goalposts. They say, do not rule it out just because it feels inhospitable. Humans borrow these lessons every day without always noticing. A DNA copying enzyme from a heat-loving microbe made modern genetic tests fast and reliable. It shrugs at high temperatures that would ruin most proteins, which is exactly what is needed to cycle through the heating and cooling of PCR. Pigments from radiation-resistant organisms are investigated as natural sunscreens and antioxidants. Enzymes from cold-adapted microbes make laundry detergents that work in cool water. Microbes that tolerate solvents and extremes find work in cleaning up oil spills and industrial waste, chipping away at pollutants that ordinary bacteria will not touch. Evolution's solutions are not only clever, they are also transferable. There are limits, and that is part of the wonder. Life needs a solvent, on Earth it is water, and a way to keep information stable while still allowing change. Too hot, and molecules fall apart faster than any repair can keep up. Too cold, and diffusion slows to a crawl that starves reactions. Too dry, and even the best vitrification eventually lets damage accumulate. Extremophils trace the edges of the possible and show that the edges are ragged, not a neat circle. For every new environment we test, life seems to find a foothold, then a strategy, then a community. Perhaps the most striking takeaway is philosophical. The phrase harsh environment assumes a baseline suited to us. To a thermophil, our body temperature is a chilly inconvenience. To a halophil, fresh water feels dangerously dilute. To a tardigrade in a ton, the vacuum of space is not home, but it is survivable, and sometimes survivable is enough. Life's habit is not to dominate everything, but to persist, to find workarounds, to endure. Where energy differences exist, light meeting pigment, minerals meeting water, heat meeting membrane, life tests the gap. Often, it crosses. So when you hear boiling acid, freezing ice, or radiation, think less of red lines and more of frontiers. Somewhere, a microbe has tuned its machinery to that key. Somewhere a tiny animal has a button it can press to become almost nothing until almost everything returns. The limits of biology are not erased by these stories, but they are pushed outward, giving us a larger, stranger, and more forgiving definition of where living things belong. Inside each cell, the genetic library is longer than you'd expect and tucked away far more neatly than seems possible. Take the DNA from a single human cell, and you could lay out about 2 meters of it end to end, yet the nucleus that holds it all is smaller than a dust mote. The trick is elegant origami. The double helix wraps around protein spools called histones to form nucleosomes, like thread-around beads. Those beads on a string coil into thicker fibers, which loop and fold again, guided by architectural proteins that clip distant segments together. The result is chromatin, part flexible, part scaffolded, organized into neighborhoods that keep certain genes close to the machinery that will read them and tuck others out of the way until they're needed. It is not just storage, it is choreography. Where a piece of DNA sits in this three-dimensional city helps decide whether it will be silent, whispering, or singing at full voice. Scale the idea up and the numbers turn playful. If you stretch the DNA from all your cells into a single line, it would reach from Earth to the Sun and back hundreds of times. Fortunately, life does not do business in straight lines. Instead, information is kept local. A gene near the nuclear pores can be transcribed and shipped out quickly. A gene sequestered deeper inside may wait its turn. Sections loop out to touch enhancers, short stretches of DNA that act like volume dials. Proteins land on these enhancers and bend the DNA so an enhancer miles away, in molecular terms, brushes the promoter of its target gene. A little contact, and the transcription machinery finds its place and begins copying RNA. No one shouts. The nucleus is a murmur of meetings. Only a small fraction of the genome directly encodes proteins, on the order of about 2%. The rest is not junk, it is context. Introns interrupt genes and are spliced out, but the way splicing occurs can change the final message, letting a single gene yield multiple proteins with different endings, or skip a section for a variant job. Long non-coding RNAs scaffold protein complexes, or help bring distant DNA regions together. Small RNAs, microRNAs and their cousins, act like post-it notes that tell the cell to degrade certain messages, or to read them more slowly. Vast tracts of sequence serve as switches and dimmers, promoters, enhancers, silencers, that control when and where a gene turns on, for how long and how strongly. In development, that control writes the body plan. A gradient of one factor turns on the first set of genes in a limb bud. Those genes turn on others. Boundaries sharpen, fingers form. The blueprint is as much timing and balance as it is parts. Layered on top of the letters sits epigenetics, marks that do not change the sequence but change how it is read. Methyl groups can be added to DNA at certain sites, and generally quiet nearby genes. Histone proteins sport tails that accept chemical tags, acetyl, methyl, phosphate, that alter how tightly the DNA is wrapped, which in turn changes access. Chemical weather outside the nucleus, nutrition, stress, sleep, toxins, can influence these markings. Some fade when eggs and sperm reset their genomes for a new generation. Some persist, carrying a faint echo of life's circumstances forward. Epigenetic regulation is why identical twins can drift apart in traits over time despite having nearly the same DNA sequence, and why the same genome can create a neuron, a muscle cell, and a skin cell simply by reading different chapters. Reading itself is precise. When a gene is active, enzymes unwind just enough local DNA and assemble an RNA copy. That RNA leaves through nuclear pores to the cytoplasm, where ribosomes translate its codons, three-letter words, into amino acids that are linked into a protein. But even here the non-coding regions weigh in. Untranslated flanks at the ends of RNAs determine how long a message will last before being recycled, and how readily ribosomes will grab hold. Regulatory RNAs can park on a message and block it, or recruit proteins that slice it up. The letters may be A, T, G, and C, but punctuation and margin notes make the meaning fluid. Hidden within the text are older stories. A surprising portion of the human genome is a scrapbook of ancient viral encounters. Long ago, retroviruses slipped their genetic material into the DNA of germ cells, sperm or eggs, and, by doing so, hitched a ride into the next generation. Many of these sequences mutated into inactivity, fossilized within our chromosomes. Others were adopted. One viral gene, now called syncytin, helps fuse cells in the placenta so nutrients can flow from parent to fetus. Bits of ancient virus serve as regulatory switches that nearby human genes now obey, and some are repurposed to tame other viruses by trapping them in molecular stalemates. What began as infections became infrastructure, proof that evolution is as much recycler as designer. The genome also holds mobile elements, transposons, that can copy or cut and paste themselves around. Most are quiet, kept in check by epigenetic locks, but during development or stress some stir. While their movements can cause mischief, they also create novelty, new regulatory sites, new splice options, new gene fusions that selection can test. Over geologic time, transposons have helped redraw the regulatory landscape, giving species different ways to use similar genes, like musicians improvising on a shared theme. Even the housekeeping of DNA shows intention. Telomeres cap the ends of chromosomes with repeating sequences, protecting genes from fraying during replication. An enzyme called telomerase can extend those caps, active in stem cells and damped in most mature tissues. DNA repair systems patrol constantly, fixing mismatches, clipping out damage from ultraviolet light, and knitting double-strand breaks back together. When repair can't keep up, cells pause division to try again. If failure looks likely, they self-destruct rather than pass along errors. The genome is not brittle parchment. It is living material with maintenance crews. The language inside is flexible enough to talk to itself. Genes can encode proteins that regulate other genes, forming feedback loops and feed-forward motifs that whisper stability into noisy chemistry. Some proteins seek out the very genes that made them to keep production steady. Others shut down their own expression when a threshold is reached, preventing runaway growth. Groups of genes fire together in circuits that toggle between states, turning a stem cell into a neuron or a lymphocyte into a plasma cell with a decision that, once made, is remembered through cell divisions. Because so much meaning sits in regulation, small tweaks can yield large effects. A change in a protein coding region might alter a single tool. A change in an enhancer can alter when and where that tool is used, sometimes with far bigger consequences for form and behavior. This is one reason closely related species can look and act very different despite sharing many of the same genes. It is also why the study of genomes now spends so much time mapping the switches in the 3D neighbourhoods, not only the list of parts. Taken together, the picture is generous. The genome is a lettered thread, yes, but also a living script performed in a particular stage set with lighting cues, props, and ensembles. 2 meters folded into a tiny room become a library with quick access shelves and deep stacks. Only a small slice writes tools. Most of the rest sets the tempo and the score, and in the margins the ghosts of old viruses and the footprints of wandering elements remind us that the text has many authors. What you are hearing as you breathe and think is that language in use, pages opening and closing, passages bookmarked, a handful of motifs returning at the right moment so a person can grow, adapt, remember, and remain themselves. 2 people can share the same genome and still take different paths. Identical twins begin with matching DNA, yet their lives quietly push those genes to perform in different ways. One twin might spend more time outdoors, sleep a little better, catch a different childhood virus, choose a different sport, favour different foods, or carry more stress. Those small divergences add up. When researchers compare twins across years, they see widening differences in which genes are active and which are kept quiet, especially in tissues that respond strongly to daily life: immune cells, fat and the brain. The letters are the same, the reading of them is not. The levers behind this flexibility belong to epigenetics, a set of molecular switches and dials that influence how genes behave without changing the DNA sequence. Picture the genome as a library. Epigenetic marks are the bookmarks, sticky notes, and shelf placements that guide what gets read and when. One common mark is DNA methylation, a tiny chemical tag added to certain cytosines that tends to hush nearby genes. Another is the modification of histone proteins. Acetyl groups loosen DNA from its spools to invite reading. Methyl groups can either open or close depending on context. These tags are placed and removed by enzymes that listen to signals from the cell's environment: hormones, nutrients, inflammatory molecules, neural activity. A burst of exercise, a period of grief, a change in diet. Each can nudge the pattern a little, like adjusting the lighting on a stage so different actors take focus. The effects show up in places you might not expect. Learn a skill and neurons strengthen particular synapses. The genes that build those connections see their chromatin open more readily, while others are tamped down. Encounter a pathogen and immune cells remodel their epigenetic landscape to respond faster the next time, a phenomenon sometimes called trained immunity. Adipose tissue, much more than a passive storehouse, adapts to feast and famine by reprogramming gene activity that controls insulin sensitivity, heat production, and inflammation. Even circadian rhythm leaves signatures. Consistent sleep and light exposure keep clock genes cycling with crisp amplitude, while irregular schedules blur the pattern, and the blur can spill into metabolism and mood. Not every signal is external. Development itself is a long epigenetic negotiation. In the earliest days after fertilization, most epigenetic marks are wiped clean so the embryo can start fresh, but not all. A special set of genes is imprinted, carrying parent of origin marks that ensure only the maternal or only the paternal copy is used. X chromosome inactivation in many cells of those with two X chromosomes is another epigenetic masterstroke. One chromosome is silenced early and stays that way through countless cell divisions, a patchwork of activity that can even be seen in the calico patterns of some animals' coats. Later, as tissues specialize, distinct epigenetic landscapes lock in cell identity, so a liver cell stays a liver cell and does not drift toward neuron or muscle. The stability is impressive, but it is not absolute. Injury, chronic stress, and aging can loosen old patterns and invite new ones. Can life's experiences reach forward into the next generation? In plants and many animals, the answer is clearly yes for certain traits. Epigenetic marks can ride through meiosis and influence offspring. In mammals, including humans, the picture is more nuanced because of those early reset phases that scrub marks. Yet a few roots remain plausible. Chemical cues from a pregnant person's body flow around the developing fetus and can shape the epigenetic setting of organs that are forming. Nutrition, stress hormones, and inflammation act as weather on that developing landscape. Sperm and eggs also carry small RNAs and a subset of chemical marks that can, in some cases, adjust gene expression after fertilization. Studies of famines and other intense environmental changes suggest that early life conditions can leave echoes detectable in children and grandchildren, subtle shifts in metabolism, growth, or disease risk. The strength and specifics are still being mapped, and not every reported effect holds up, but the direction is consistent. Biology remembers more than the sequence alone. None of this turns destiny into putty. Epigenetics is plastic but not magical. A methyl group on a gene associated with muscle strength will not make a person sprint without training. A loosened chromatin region near a learning gene still requires practice and sleep to cement memory. Likewise, some aspects of identity are anchored deeply in inherited sequences, with epigenetics providing fine-tuning rather than wholesale rewrite. The interplay is exactly what makes living systems resilient. Enough stability to keep a body coherent, enough flexibility to adapt without starting over. Daily habits are where these molecular details meet ordinary life. Physical activity shifts muscle and liver epigenomes toward better glucose handling and improved mitochondrial function. Some of these changes persist for weeks, making it easier to maintain the habit once begun. Diets rich in varied plants deliver fiber and phytochemicals that the microbiome converts into short-chain fatty acids, acetate, propionate, butyrate, which enter cells and can directly influence histone acetylation, opening chromatin around anti-inflammatory genes. Chronic stress, if unrelieved, can bias marks near genes that govern the stress axis, keeping the system on a hair trigger. Practices that improve sleep, social connection, and recovery tilt those marks back. Sunlight, through vitamin D pathways, talks to immune cell chromatin. Pollution can etch less helpful marks that raise risk. The signals are gentle, cumulative, and often reversible, which is why small, steady choices matter so much. Twins highlight the balance beautifully. Start with two nearly identical genomes. Over childhood, their environments diverge. Different friends, different colds, different sports, different teachers. By adulthood, their patterns of methylation and histone modification no longer match point for point, and those differences correlate with traits as varied as body composition, immune responses, and even preferences. If they later live together for a time or adopt similar routines, some differences shrink again. The genome is the score, epigenetics is the performance, responsive to the room. There are also cases where epigenetic misfires contribute to disease. If marks silence a tumor suppressor gene or activate an oncogene at the wrong time, cell growth can slide out of control. Some cancers carry few mutations but many epigenetic abnormalities. In those, drugs that inhibit enzymes that lay down or remove marks can restore a safer expression pattern. In autoimmune disorders, immune cell epigenomes may be tuned too far toward attack. In metabolic disease, chromatin around genes that control appetite and insulin sensitivity can be set to a less favorable state. The same plasticity that supports adaptation can, under chronic insult, drift toward trouble. One reason prevention and early course correction are powerful. A good way to think about inheritance and identity is musical. DNA writes the key and the available notes. Epigenetics sets the tempo, the dynamics, and which instruments come in at which bars. Lifestyle and experience are the conductor's gestures, never rewriting the notes, but deciding how they are played tonight in this hall for this audience. Some motif from last season may linger, a faint echo carried by marks in germ cells or by the chemistry of pregnancy. But most of the performance is freshly interpreted. That is comforting. It means identity is neither rigidly pre-written nor blown entirely by the wind. It means small experiences can leave molecular bookmarks, reminders to open certain pages more easily, to skim others, and those bookmarks can be moved again. So when two people share a genome and diverge, or when one person changes course and feels their body follow, the explanation runs through quiet chemistry. Tiny tags on DNA, subtle tweaks to spools, loops that bring distant phrases together. A life is read, not just encoded. The way it is read shifts with light, with food, with work, with love, with rest. And sometimes, a trace of that reading, like the gentle dent of a well-used page, carries on to the next chapter. Evolution is a backstage tinkerer, not an architect with blueprints. It works with what is already on the workbench, bones, enzymes, nerve circuits, and modifies, repurposes, trims, or duplicates as circumstances change. A fin can begin as a leg. A feather can start as insulation and end as an airfoil. A protein that once cleared household clutter in the cell is recruited into the lens of the eye because it happens to be transparent and stable. This is bricolage, problem solving by clever reuse. The result is not a march toward perfection, but a branching, meandering map of solutions that were good enough in their time and place. Nowhere is the tinkerer's touch clearer than in the history of Wales. Their ancestors were hoofed land mammals that trotted along riverbanks in the early chapters of their story. You can still see hints of that ancestry in fossil ankles shaped for running, and in teeth meant for chewing, not sieving krill. Step by step across millions of years, those lineages slid back into the water. Early forms had long tails and stout limbs with webbed feet. Later ones stretched their spines and flattened their tails into powerful flukes. Front legs became paddles with tightly packed finger bones. Hind legs dwindled, and in modern whales, survive as tiny pelvic bones buried in muscle, now used to anchor reproductive organs rather than to walk. Even the nose migrated. What began at the tip of the snout crept upward along the skull to become the blowhole, a neat fix that lets a whale surface, breathe, and dive without lifting a heavy head. The ear tells the same tale. Sound travels differently in water than in air, and land ears do not work well submerged. Whale skulls evolved dense, isolated ear bones and fat-filled sound channels that funnel vibrations to inner ears tuned to aquatic acoustics. Later, some lineages added echolocation. A forehead organ called the melon focuses clicks. Returning echoes paint acoustic pictures in the dark water. At every step, nothing appears from nothing. Old parts take on new jobs. Muscles and bones are reweighted and rewired. The toolkit stays familiar even as the body's purpose shifts from wading to sprinting through the sea. Birds are another masterpiece of modification. Their story begins with small feathered theropod dinosaurs, fast two-legged hunters with grasping hands. Feathers arrive before flight, likely as insulation, display and balance aids for running and leaping. Over time, those feathery veins broadened and aligned, arms lengthened, and the shoulder joint rotated to permit powerful downstrokes. Air sacs invaded the skeleton, making bones lighter and weaving a through-flow breathing system that is astonishingly efficient. The wishbone, fused collarbones, acts like a spring during each wing beat, storing and returning energy. Claws on forelimbs shrank in most lineages, tails shortened into a pygostyle that anchors steering feathers. Nesting behavior and brooding, seen in fossils of dinosaurs curled around clutches, glide seamlessly into bird life. Feather first, flight later, balance first, wing beat later. Again, purpose is stack rather than replace. A structure shaped for warmth becomes a surface for lift, and a running leap becomes a glide, then powered flight. Both stories show what evolution is and what it is not. It does not set targets decades or eons ahead. It makes local improvements under current constraints. That is why living designs carry odd detours. A nerve that runs from the brain to the larynx takes a long loop down into the chest and back up. Efficient enough in fish, where the route is short but comically long in giraffes. Replumbing the neck from scratch is not on the table. The nerve elongates as necks lengthen, a workable compromise born of history. Eyes assemble from parts available. Light-sensitive proteins borrowed from ancient chemistry. A patch of skin that invaginates. A clear dome that happens to focus. The end result can be exquisite, but the steps are incremental and each is useful on its own. Light detection before imaging, dim shapes before sharp pictures. Tinkering thrives on a few simple tools. One is duplication. When a gene is copied, one version can keep the original job while the spare is free to drift, picking up a new function. Entire families of hemoglobin variants, odor receptors, and digestive enzymes owe their diversity to this make a backup, then experiment strategy. Another is regulatory rewiring. A limb gene can be turned on earlier or later, in more cells or fewer, at the front of a limb bud or the back, producing fins, fingers, elongated digits, or lost ones without changing the core protein. Much of the difference between closely related species lies not in the parts list, but in the timing and dose of those parts. Convergence is a recurring punchline. Put unrelated lineages in similar conditions and the tinkering points them towards similar solutions. Sleek, torpedo-shaped bodies appear in sharks, tunas, dolphins, and extinct ichthyosaurs. Gliding membranes stretch between limbs in flying squirrels and sugar gliders. Camera-style eyes evolve independently in vertebrates and cephalopods. The paths differ. The physics is the same. The workshop yields the same broad answer with different fasteners. Because there is no single ladder upward, primitive and advanced are poor labels. Horses did not progress neatly from many toes to one. They sprouted and pruned branches as climates changed, with multiple body plans overlapping in time. Crocodiles look ancient because their niche stayed stable. They are not stuck, they are successful. Bacteria did not get left behind in an earlier act of the play. They dominate the biosphere precisely because their strategies, fast reproduction, gene swapping, metabolic versatility, work in almost any scene. Progress suggests a finish line. Persistence recognizes that the race is to keep running through droughts, ice ages, volcanism, and asteroids. The fossil record is full of transitional forms not because evolution is uncertain, but because every lineage is perpetually in transition. A jaw joint that slowly migrates to the middle ear, a fin with bones that look suspiciously like a wrist, a feather that stiffens along one edge. Each intermediate is complete and competent for its own way of life. Natural selection tests small changes in real time. The ones that help leave more copies, the ones that hurt fade, the neutral ones drift with luck. Over deep time, the pile of small edits reads like a novel with long chapters and many subplots. Even loss can be a kind of gain. Cavefish shed eyes when darkness makes eyes expensive luxuries. The savings go into other senses. Parasites streamline genomes and bodies when hosts provide tools they no longer need, trading independence for efficiency. Flightless birds on predator-free islands put energy into speed on land and size instead of wings, a smart swap until new predators arrive. What looks like backsliding is often just accounting. Evolution's tinkering also means that solutions come with trade-offs. A peacock's tail impresses mates but slows escapes. Human bipedalism frees hands for tools and gestures, but strains the back and rearranges the birth canal. Sickle cell traits increase malaria resistance while risking anemia when inherited in the wrong combination. There is no free upgrade, only balances that make sense in a given context. At heart, the process is simple. Variation arises through mutation, recombination, duplication, environments filter, through climate, competition, chance. Survivors pass along what worked. Repeat across generations and patterns appear. When seas invade continents, coastal animals with a taste for wading and a knack for holding their breath do better. A few million years later, some descendants no longer need land at all. When small, feathered dinosaurs explore trees and open air, those with slightly broader feathers and a stronger shoulder survive leaps better. Eventually, some are birds. When conditions swing, lineages split, radiate, stall, or end. The winners are not the most complex or the most beautiful. They are the ones whose parts could be retuned for the next song. So evolution is not a staircase. It is an improvisation that never stops. It keeps old motifs, introduces new instruments, and sometimes brings back a theme with a twist. Whales carry legs that forgot how to walk but remember how to anchor. Birds wear feathers that warmed their ancestors before they lifted their descendants. And life, taken as a whole, is not aiming higher so much as it is learning, again and again, how to stay. A flower is a promise written to a particular audience. Petals shape the landing pad and the runway. Scent drifts on air currents, timed when visitors are awake. Nectar sits like a tip jar at the end of a narrow corridor that only a correctly built tongue can reach. Pollinators return the favour by arriving with the tools the plant has quietly requested. Hummingbirds hover before red tubular blossoms that match the length of their bills, trading sugar for pollen placement on foreheads and throats. Bees prefer blues and yellows and see ultraviolet patterns invisible to human eyes, nectar guides that point like runway lights to the prize. Night-blooming cacti open pale flowers that reflect starlight and smell of fruit. Bats arrive as the lights go down, faces dusted with pollen like miners in a sweet cave. Even wind-pollinated grasses have played a role in this grand negotiation, shedding the petals they no longer needed to focus on producing clouds of powder that drift to equally stripped down flowers. Everywhere, form follows partner. Some of these relationships are so specific they read like riddles solved across generations. When Charles Darwin was sent a long-spurred orchid from Madagascar, he predicted a moth with a tongue to match, nearly as long as a forearm, must exist to reach the nectar at the bottom. Decades later, the moth was found, tongue exactly as forecast. Fig trees and their tiny wasps are equally intertwined. Each fig species hosts its own wasp, which crawls into the fig's hidden flowers, pollinates them, lays eggs, and dies. The wasp's offspring mature inside, then carry pollen away to the next fig. Failures occur, cheaters appear, storms disrupt schedules, yet over a long time the fit tightens. Coevolution is an arms race only when there are enemies. Between partners it is a duet, one part testing, the other applying, until the melody locks. Underwater the choreography looks different but follows the same logic. Sea anemones bristle with stinging cells primed to harpoon anything that brushes by. Clownfish somehow slip among the tentacles unhurt, their skins coated in a mucus that the anemone does not recognize as food or foe. The relationship takes practice. A new clownfish performs a careful acclimation ritual, touching and retreating until the anemone accepts it. Once accepted, the fish patrols the area, chasing away butterfly fish and other nibblers that would chew the anemone's delicate tips. It fans the tentacles, improving water flow, and its waste fertilizes its host with nitrogen compounds that boost growth. Small payments that add up. In return, the anemone's forest of stings turns into a fortress. Predators that would swallow a clownfish whole cannot risk a mouthful of nematocysts, and the fish live longer, breed more, and pass on the habit. Clownfish societies add another layer. They live in size-ranked groups with a dominant female at the top, a breeding male next, and several non-breeders below. Should the female die, the breeding male changes sex and takes her place, and the larger subordinate moves up. The anemone is not just an apartment, it is the stage on which this flexible social ladder plays out. Remove the anemone and the caste dissolves. Protect it, and the hierarchy and the mutualism stays stable for years. Even here, cooperation has checks. If too many fish crowd the tentacles, juveniles are chased off. If the fish population dwindles, the anemone's growth slows. Balance keeps the bargain prosperous. On bare rock, where hot, cold, dry or salty conditions would defeat most plants, lichens become the first settlers. Look closely, and a lichen is not a single organism but a partnership. A fungus builds the house, and a photosynthetic algae, or cyanobacterium, stocks the pantry with sugars made from light. Many lichens actually host more than two partners. Bacteria living in the thallus contribute vitamins or nitrogen, and the fungal partner can be a duo. The structure is modest, crusty, leafy, or branching forms that look like paint, ruffles, or tiny shrubs, but its competence is impressive. Lichens mine minerals from rock acids, catching dust and dew, slowly making the first thin soils that future plants will root in. They break down with the patience of a century, then scatter through spores or little packets called soredia that contain both partners, ready to start again wherever the wind lets them fall. Because the lichen's photosynthetic partner is sensitive to air chemistry, lichens also serve as living gauges of pollution. In cities where nitrogen oxides are high or sulfur compounds still haunt the air, delicate species vanish, clean up the atmosphere, and they return in variegated patches on trees and stone. For all their endurance, they maintain a simple rule. The fungus must build and protect, the alga must feed, the whole must tolerate swing after swing of wet, dry freeze thaw. If one partner falters, the result is not a clean split, but a quiet fade. Their long, slow lives remind us that cooperation can be unhurried and still powerful. Once you look for partnerships, they multiply. Cleaner wrasses set up stations on reefs where large fish line up to have parasites plucked from their skin and gills. The clients hold still and open their mouths while the cleaners dart in and out. The service reduces disease and improves swim efficiency. The payment is food for the wrasses. Leaf-cutter ants farm fungi underground, feeding them chewed leaves they cannot digest on their own. The fungus breaks the plant matter down, and the ants harvest the swollen tips as food. The ants carry bacteria on their bodies that secrete antibiotics to fight rival molds, a three-way alliance of farmer, crop and chemist. In grasslands, some plants house nitrogen-fixing bacteria in root nodules, paying with sugars in exchange for ammonia that the plant weaves into proteins, fertilizer made in-house without a bag or a tractor. Even partnerships with risks can persist when the ledger stays favourable. Oxpeckers riding large mammals sometimes remove ticks, a clear win, but they also sip a little blood and may keep wounds open, a cost. Many hosts tolerate them anyway, especially when ticks are thick and the extra picking pays. Corals, which rely on photosynthetic partners for much of their daily sugar, eject those algae during heat stress, bleaching to survive the moment at the expense of long-term health. If cooler water returns, they can re-establish the relationship, sometimes with strains more tolerant of heat. The mutualism rewrites itself to fit the weather. Cooperation does not erase competition, it redirects it. Flowers compete for pollinators, but they also specialize to spread out the workload. Different colors, shapes, and bloom times reduce overlap. In the soil, mycorrhizal fungi link a stand of trees, and while individuals may jockey for light above, the network below smooths the feast and famine by shuttling carbon and water to where short-term need is sharpest. The net effect is a forest that weathers drought and pest waves better than a collection of isolated trees would. The presence of a partnership can change the playing field so thoroughly that new species move in and a community reassembles around the benefits. The reason these arrangements keep appearing is that they solve real problems. Energy is hard to capture. Partners that can trade sugars for nitrogen, or shelter for defense, or transport for precision do better than loners when conditions wobble. The living world is a marketplace where trust is built with molecules, enforced by feedback, and revised when circumstances shift. Some deals span moments, like a bee visiting a blossom. Some last lifetimes, like an anemone sheltering its fish. Some last geological ages, like fungi and algae painting stone. Step back and the story that looked like rivalry becomes a tapestry. There are predators and prey, yes, and there are races to bloom first, to grow fastest, to hold a territory. But the scaffolding of ecosystems, the parts that keep energy flowing, waste recycling, soils forming, bodies fed, leans on partnerships tuned across time. Life gets ahead together more often than it gets ahead alone. That is not sentiment, it is accounting. Cooperation is not a soft alternative to selection, it is one of selection's best tools for surviving a complicated world. Each inhale is a small history lesson. The oxygen sliding into your lungs was stitched into the air by living things, by leaves that split water with sunlight, and by microscopic drifters in the sea that do the same in glass-clear silence. In spring and summer, satellites can watch entire continents green and the planet's carbon ledger tilt as forests draw down carbon dioxide and release oxygen. In fall and winter, the balance eases back as leaves rest or fall and microbes return stored carbon to the air. The ocean breathes too. A thin sunlit skin at the surface swirls with phytoplankton, diatoms with glassy shells, cyanobacteria the size of dust, fixing carbon, releasing oxygen, and feeding nearly everything that swims. Tiny grazers eat them, fish eat the grazers, and the leftovers snow downward as marine flakes, a slow blizzard called marine snow that helps ferry carbon to the deep. In this living tide, certain places punch far above their area. Coral reefs, for example, occupy well under 1% of the ocean, yet host roughly a quarter of known marine species. The math works because reefs are both cities and factories. Corals are animals that farm sunlight. Each polyp harbours photosynthetic partners that pay in sugars by day, while at night the coral reaches into the current to catch passing plankton. Layer by layer, corals lay down limestone skeletons that break waves, sculpt courtyards for fish, and shelter a menagerie of invertebrates. The shape of the reef, ledges, grottos, pinnacles, creates microclimates of light and flow. Algae carpet some zones and feed grazers, sponges pump and filter, plumbing the water and recycling nutrients. Cleaner wrasses set up service stations where large fish queue to have parasites plucked from their gills. A reef is not just biodiversity stacked in space, it is speed. Molecules are used, reused, and handed off so quickly that little leaves the neighbourhood. On land, the engine of renewal often sounds like crackle and pop. Fire, in the right dose and season, is not destruction, but reset. In prairies, flames clear last year's thatch, warm the soil, and release a pulse of nutrients that grasses rapidly reclaim. Many pine forests are built on this rhythm. Some pines seal their cones with resin that only softens in heat. A blaze opens them like popcorn, and seeds fall into a bed of light and ash. Thick bark protects the mature trunks, and seedlings surge while competitors are briefly absent. When fire is excluded for too long, fuel piles up, and later burns run too hot, scalding the very tissues meant to survive. The lesson is not that fire is good or bad, it is that ecosystems have pulses, and fire is the metronome for many of them. After the flash comes the feast. Decomposition is where life does its clever bookkeeping. Fungi send hyphae into logs and leaf litter, secreting enzymes that unlock lignin and cellulose, the armoured sugars of wood. Bacteria finish what fungi begin, clipping molecules down to pieces plants can use again. Detritivores, woodlice, millipedes, springtails, shred the big into the small, and earthworms churn the mix into soil with stable crumbs that hold water and air. Even bones return to circulation. Beetles specialise in carrion, birds and mammals clean a carcass in hours, microbes dissolve the rest. Nutrients that once stood as trunk, hoof and horn find their way into moss and meadow. There is pathos in death. There's also continuity. The carbon in your breath today will be part of a leaf in summer. The calcium in a fallen antler may become the shell of a snail. Predators write another kind of rhythm, one measured in restraint. Where sea otters thrive, they eat sea urchins, which spares kelp forests from being grazed to nubs. In the shade of kelp, fish nurseries bloom, the water stills, and carbon sinks as fronds fall and drift to deep sands. Remove the otters and urchins pulse outward, sculpting urchin barrens. On land, wolves tamp down elk browsing in river valleys. The riparian willows rebound, beavers return to build ponds that slow floods and store water through dry months. These are the famous examples of trophic cascades, but they are not rare miracles. They're reminders that fear and appetite travel through ecosystems as surely as molecules do. Predators do not just remove bodies, they edit behavior, opening space for plants to grow and engineers like beavers to work. Waterways knit land and sea and carry pulses that tie distant places together. Salmon hatch in cold streams, grow at sea, then swim back to their birth waters bearing the ocean's nitrogen and phosphorus in their bodies. When they die, bears, eagles and microbes redistribute that marine wealth through forests. Needles green a little brighter, moss thickens, invertebrates surge. Floodplains tell a similar story. When rivers spill, they leave silt and seed in their wake. When they retreat, backwaters warm and become cradles for fry and frogs. Dams and levees that tame one risk can pause these gifts. Reconnection brings the pulse back. The ocean has its own seasonal and structural beats. Upwelling coasts see deep, nutrient-rich waters rise when winds push surface layers aside. Phytoplankton answer, with blooms so dense they tint the sea from space, and fisheries blossom in step. Anchovies, sardines, the predators that chase them. In the open ocean, floating deserts give way to oases around seamounts where currents lift nutrients and life concentrates. Even the microbial loop quietly stitches scraps back into useful form. Bacteria absorb dissolved organic carbon, tiny predators eat bacteria, larger plankton eat them, and the carbon re-enters the classic food web instead of drifting uselessly away. Because energy is precious, life learns not to waste. Many seeds are packaged with fruits that invite animals to carry them off and deposit them, pre-fertilized, in new ground. Dung beetles roll, bury and seed future grass with their labour. Termites build air-conditioned mounds that capture breezes and regulate humidity, so the fungal farms inside keep perfect time. In deserts, shrubs nurse seedlings in their shade. Leaf litter beneath them holds more moisture and feeds soil life that in turn feeds roots. A savanna is not merely scattered trees and grass. It is a lattice of facilitation, linking plants, fungi, insects, and large herbivores across space. At the planetary scale, these local exchanges sum to cycles. Carbon moves from air to leaf, to soil, to air again. Nitrogen is fixed from the sky by microbes, built into protein, then returned by fire, feces, and fungi. Phosphorus rides weathered rock into streams and seas, feeds plankton, settles to mud, and climbs up in the bones of fish and the beaks of birds that return to nest on cliffs, guano delivering phosphorus back to land. The loops are not perfect circles, they meander and pool, but the grand pattern is reuse. Waste is a concept for closed minds, not for open ecosystems. Even disturbances that look like breaks often become beats. A hurricane opens forest canopies so light reaches the floor. Pioneer species rush in, insects arrive, birds follow, and over years the forest layers itself back together with new mosaics of age and height. A fallen tree bridges a stream, shifting currents, making eddies where silt gathers and caddisflies thrive. A dead whale that sinks becomes a banquet on the abyssal plain, first for sharks and hagfish, then for bone-eating worms and sulfide-loving bacteria. Each end writes opportunities in a different script. When you breathe now, part of the oxygen is a gift from a leaf that opened its pores this morning, and from plankton that split water in a sparkling top metre of sea. The same breath will leave us carbon that, given time, could be woven into a blade of grass, a spruce needle, a diatom shell. Ecosystems are not machines humming toward a finish line. They're choirs keeping time, passing notes from voice to voice, filling gaps when one singer rests. The pulse is everywhere, wind through kelp, sap in xylem, mice under snow, fungi through loam, and everything that lives plays an instrument, including you. Push a spade into a patch of garden and you're not just turning soil, you're meeting a workforce. Earthworms thread tunnels through the dark, swallowing crumbs of leaf and wood, mixing them with grit and microbes, and returning the blend as castings richer than what went in. Each cast is a tiny fertilizer pellet, more available nitrogen, more phosphorus and potassium, and a feast of microbial life ready to pass nutrients along to roots. As they move, worms stitch the ground together. Their mucus glues particles into stable crumbs that hold both air and water, so rain sinks instead of skating off, and roots find pockets of oxygen where they can breathe. The burrows themselves are plumbing, carrying stormwater deeper and easing floods, and highways, letting roots follow the path of least resistance to new territory. Even seeds hitch rides. Some fall into tunnels and germinate with a head start, tucked in moist walls where competition is lighter and the microclimate is kind. Worms don't work alone. Their guts are meeting halls for bacteria and fungi that finish breaking tough molecules into plant-ready forms. On their way through, microbes swap genes and tools, sharpening the community's ability to digest whatever the season offers. When a worm dies, the nutrients tied up in its body return to the cycle. Nothing in the system lingers unused for long. In fields with active worm populations, water infiltration improves, compaction eases, and crops weather dry spells with less complaint. In forests, castings pepper the leaf litter like slow, generous raindrops of fertility. The story is not glamorous, but it is foundational. Fertility is engineered from the bottom up by animals that never see the sun. Rise from the garden and follow a stream into the trees, and another builder appears with bigger blueprints. Beavers are engineers with teeth and patience. They fell saplings and drag them to running water, weaving branches with mud and stones until the current slows and fans into a pond. The dam is not a wall as much as a sieve. Water leaks through and over but gently, and that gentleness changes everything. Sediment drops out to the bottom where it would have rushed away. Nutrients settle and feed algae and aquatic plants that never had a foothold in the scouring flow. As the pond spreads, it creates a mosaic. Open water, marsh, soggy meadow, willow thicket, each niche a different invitation to insects, amphibians, birds and mammals. Dragonflies write calligraphy over the surface, ducks make landings, frogs chorus from the edges, muskrats build lodges that echo the beavers in miniature. The changes travel downstream and upslope. Slower water means fewer floods and more steady flow through dry months. Ponds store rain like batteries and release it when creeks would otherwise shrink to a trickle. Groundwater tables rise as the pond's pressure pushes water sideways into the soil, recharging springs and wells. Fire risk can drop where wet meadows stretch like green fire breaks through the forest. Even temperature moderates. Deep pools stay cooler in summer, giving cold water fish a refuge. When a dam fails or a family moves on, the pond silts into a rich meadow that captures carbon in peaty layers. A century later, a line of willows marks where water once stood. Given the chance, beavers do more than make ponds. They reset hydrology, redraw habitat maps, and leave a scaffold on which other species build. Travel farther, out to sea, and the architects shrink again, this time to the size of a pencil eraser. Coral polyps look like tiny sea anemones wearing limestone collars. They live in vast colonies, each animal secreting calcium carbonate beneath itself, one thin layer at a time. The partners inside their tissues, photosynthetic algae, pay them in sugars spun from sunlight, and the corals pay the rent in carbon dioxide and a well-lit apartment near the surface. Over years and then centuries the colony rises and spreads. Storms break branches, which reattach and grow into new heads. Rubble gathers and cements. Fish graze the film of algae so it does not smother young corals, and sponges pump seawater, digesting microscopic scraps that would otherwise drift away, turning waste into nutrients the community can use. Wave by wave, tide by tide, the limestone mass becomes a reef that can be seen from orbit, a bright line where Livingstone meets open blue. Reefs are machines for turning structure into life. Ledges and crevices create homes and ambush points, shade and sun, fast water and still. This physical diversity makes biological diversity possible, and the speed of recycling keeps the party going. Nutrients come in pulses, excrement from a passing school, a drift of plankton on a night current, and are captured, chewed, excreted, and recaptured so many times that little is lost. Predators cruise, cleaners set up shop, corals grow by day on sunlight, and by night on the plankton they catch with stinging tentacles. In storms, reefs blunt waves and shield coasts. In calm weather, they raise islands by catching sand and building it into beaches. Each limestone grain is a receipt written by a polyp that lived, ate, and built. Once you start looking for builders, they are everywhere. Termites in the savannah raise cathedral mounds with internal air shafts that work as natural chimneys. Hot air rises, cool air sinks, and the colony adjusts vents to keep the interior in a narrow comfort zone that would be hard to achieve with electricity. Those mounds concentrate nutrients, and rare rains green them first. Grasses here grow taller, drawing grazers who distribute seeds and dung farther. Prairie dogs excavate towns that stitch the soil with tunnels. The digging mixes layers and creates patches of fresh growth that feed bison and pronghorn, and the burrows themselves become housing for owls, snakes, and beetles. Mussel beds on temperate shores and oyster reefs in estuaries glue themselves into rough pavements that calm waves, trap silt, and invite entire neighbourhoods of crabs, young fish, and anemones to move in. Kelp forests, built by a single species of brown alga anchoring to rock with holdfasts, stand like underwater redwoods. Their canopies slow currents, shade the seafloor, and gather life from urchins to sea lions. What unites these architects is not size or fame, but feedback. They alter their environment in ways that make it better for themselves, and, incidentally or deliberately, for many others. Earthworms make soil that grows plants that feed worms and everything up the chain. Beavers make ponds that grow willows that feed beavers, and shelters that host herons, mink, and turtles. Corals make reefs that attract grazers that keep the corals clear to keep building. This circle widens, and with it resilience. Systems with strong engineers tend to bend rather than break. Storage of water or nutrients smooths the feast-famine cycle. Nooks and layers give species escapes when weather or predators turn harsh. Even decay fits the blueprint. When a beaver dam ages and fails, the silted pond becomes a wet meadow rich with sedges and flowers. When a coral head dies, the skeleton remains as a foundation for new recruits. When an old termite mound slumps, its enriched soil grows trees faster than the plain around it. Building and unbuilding are phases of the same long project, with materials recycled, not wasted. Life's architects do not seek permanence, they seek continuity. Humans have always lived in the neighbourhoods these builders made, sometimes as tenants, sometimes as demolition crews, sometimes as apprentices. Where earthworms were absent and then arrived, farmers noticed soils loosen and yields climb. Where beavers were extirpated, rivers sharpened and valleys dried. Where they were invited back, wetlands returned and wildlife followed. Where reefs decline, coasts lose their breakwaters and fisheries their nurseries. Where reefs recover, the water clears and the web tightens again. The lesson is not only ecological but practical. To repair a place, you can often start by hiring its original contractors, protect the worms from deep compaction, give beavers room to work, ease stress on corals by improving water quality, let oysters rebuild their beds, and the quiet architect will take the project from there. So much of the planet's beauty is not accidental. It is constructed by patient hands, many tiny, some webbed, some sticky with limestone, and maintained by habits learned across deep time. A meadow's bounce, a stream's meander, a reef's lacework edge. Each is a structure with a builder's signature if you know how to read it. And under your next step, in the soil that gives and springs back, the most modest of engineers is probably already at work turning yesterday's leaves into tomorrow's green. Look closely at a leaf and you'll see a map. The midrib splits, then splits again, and again, until the paths are hair-thin and everywhere. That same branching logic guides the limbs of a tree and the drainage of a hillside. When a system has to deliver or collect something, sugar, water, sediment, oxygen, efficiently from many places to one, it tends to grow a fractal, repeating patterns that scale from large to small. In leaves, branching veins feed photosynthetic cells and provide detours when an insect chews through a line. In your body, arteries branch into arterioles and then into capillaries, so every cell sits within a few micrometers of a delivery route. In river basins, trickles gather into rills, then creeks, then rivers that snake to the sea. The angles aren't random. Branches usually meet at junctions that balance flow with minimal wasted energy. Nature's quiet engineering. Even the proportions echo across scales. A thick trunk with thinner boughs, a main channel with lesser tributaries, a primary vein with a lace of support, each echoing the same rule with different materials. Follow rivers long enough and the pattern deepens. Water cuts softer rock first, then works on the harder parts, exploring and abandoning paths until a network remains that mirrors the statistics of countless storms. Tributaries join at characteristic angles. Segment lengths follow power laws. Meanders lengthen with time, bend until a cutoff forms, and leave oxbow lakes like commas scattered across the floodplain. Put ink in a tank of corn syrup and stir, then unstir, and filaments fold back on themselves in a way that looks like river braids or lung airways. A demonstration that flow loves to laminate and layer. Your lungs use this lesson to pack enormous surface area into a small volume. Branching bronchi end in millions of tiny alveoli, frothy with capillaries so that oxygen doesn't travel far before hopping into blood. The spiral is another favourite, appearing wherever growth adds new material at a steady rate, while the size of the growing edge keeps changing. Shells expand by accretion along an ever-widening lip, and the curve that results is a logarithmic spiral. The shape stays the same while scaling up, like a song played in a higher key. Pine cones and sunflower heads arrange seeds in spiraling families whose counts often match consecutive Fibonacci numbers. New seeds slip into the least crowded gaps, and the math of optimal packing writes those counts into the pattern. In storms, spirals form because air rushing toward a low-pressure center is deflected by the planet's spin. As bands of cloud and rain wrap around the eye, the radar picture starts to look eerily like a nautilus painted in vapor. Some galaxies show sweeping spiral arms for a different reason. density waves ripple through the rotating disk of stars, gathering them into lanes that persist while individual stars wander in and out, the way cars slow and bunch through a traffic wave that no single car creates. Patterns appear even when behavior seems random. Dripping taps, heartbeat intervals, forest fires, and stock markets all flirt with chaos. Tiny differences in starting conditions can produce very different outcomes. Yet the chaos is not pure noise. It clusters around strange attractors, preferred shapes in the space of possibilities. A forest that accumulates fuel slowly and burns occasionally settles into a rhythm where small fires are common and huge ones rarer, a balance some scientists call self-organized criticality. Sand beetles do it too. Add grains one by one, and you get many little avalanches and few spectacular slides, the statistics of which repeat in a predictable way. Rivers flood, deltas shift, dunes march, unpredictable in the exact moment, patterned in the long arc. Look at an animal's coat and the geometry becomes chemical. Stripes and spots can emerge from reaction diffusion systems where one molecule activates pigment production and a second molecule inhibits it a short distance away. Depending on the rates and diffusion speeds, you get zebra bars, leopard rosettes, or the dot matrix of a fawn. Fish and seashells draw wave-like moires as pigment cells nudge neighbours into similar states, or as growth fronts push pigments out in pulses. Even your fingerprints, unique as they are, arise from waves of growth and stress that ripple across the fetal skin, turning tiny mechanical differences into whirls and loops. At smaller scales, symmetry rules for different reasons. Many viruses pack their genes inside icosahedral shells because 20 identical triangular panels make a strong closed container with minimal instructions. Snowflakes sprout 6 arms as water molecules crystallize into hexagonal lattices, each arm responding to the same temperature and humidity history, which is why they match until the air changes and the growth tips try something new. Soap bubbles and wet foam settle into hexagonal honeycombs because a hexagon tiles a plane with minimal boundary length for a given area. Less surface means less energy. Bees discover the same trick for storing honey. Build walls at angles that save wax, and the comb supports heavy loads with light materials. Life borrows geometry for function. The hexagons in a wasp's eye pack photoreceptors densely to maximize light capture. The spiral staircase of proteins in a bacterial flagellum twists at a precise pitch to translate rotary motion into forward thrust. Microtubules, the stiff struts inside cells, assemble into cylinders of 13 protofilaments, arranged so that the structure has a gentle helical bias, ideal for resisting buckling yet allowing motors to walk along their tracks. Hemoglobin packs 4 subunits into a symmetric tetramer, switching shape as oxygen binds to make pickup and delivery efficient. At larger scales, ant trails and human footpaths both approximate geodesics, the shortest paths that avoid obstacles while balancing slope and effort. Even the spaces between things organize themselves. Crack a drying mudflat and the polygon network that forms looks like cooling basalt columns or the crazing in old paint. All 3 arise from shrinkage constrained by neighbours. When tension exceeds what the material can bear, cracks pop and then stop when they meet others, leaving a patchwork of efficiency. Frost patterns feather across a window where temperature gradients steer crystal growth. Dendrites branch where the edge is coolest and starve where heat piles up, drawing stars that look like seaweed or fern fronds. Patterns bridge scales not just by appearance but by math. Power laws, the kind where doubling one quantity multiplies another by a constant factor, turn up in city sizes, earthquake energies, and the number of branches in plants. Metabolic rates scale with body size in predictable ways, because transport networks inside bodies obey the same efficiency rules that rivers do. A mouse and an elephant differ by orders of magnitude, but their hearts and blood vessels still reflect a common design code: minimize energy, reach everywhere quickly, and keep resistance low. None of this should make the world feel mechanical. The delight is in the variation around the theme. Leaves follow Venetian rules, yet a maple and a ginkgo draw entirely different maps. Spirals guide a sunflower seed packing, but wind and water and curious birds smudge the logic at the edges. A zebra's stripes are a chemical reaction frozen in fur, yet no two animals share the same barcode. Pattern and accident hold hands. Structure provides the stage and chance writes nightly improvisations. Why does nature prefer repeating motifs? Because energy, materials and time are limited. Fractals distribute resources while keeping construction cheap. Spirals allow growth without redesign. Hexagons fill space efficiently with minimal boundary. Reaction diffusion paints complexity from simple rules, saving the cost of micromanagement. The universe rewards solutions that work at many scales and adapt as they grow. Life, being practical, chooses from that toolbox again and again. So the veins in a leaf can teach you about rivers. A shell can whisper about storms in the tropics. A galaxy can hint at a sunflower. There is comfort in that echo. Atoms to organisms to landscapes, all humming the same handful of tunes, each in its own key. Some living things make their own light the way others make heat. In the dark water of a warm cove, a hand trailing through the surface wakes tiny dinoflagellates, and every swirl answers with a blue-green spark. The chemistry is tidy. A molecule called luciferin meets oxygen with the help of an enzyme, luciferase, and the reaction releases photons instead of warmth, cold light. In the deep sea, where sunlight never arrives, glow is language and lure. Anglerfish dangle a living lantern grown from symbiotic bacteria. Gulper eels flash and fold. Squid write messages across their skin with twinkling organs called photophores. On land, fireflies time their blinks like metronomes to find one another in summer fields, each species tapping a rhythm and hue that says this is me. A Hawaiian bobtail squid farms light the way a gardener tends a bed, housing bacteria in a special organ and dimming their glow at dawn to match moonlight, so its shadow disappears against the bright surface above. The trick everywhere is efficiency. Bioluminescent reactions turn chemical energy into light with very little heat. A feat engineers still envy when they build LEDs and want brightness without burn. Not all dazzling colour is made by dye. Peacock feathers shimmer because of structure. Layers of microscopic keratin and melanin granules spaced just right to scatter light in a way that boosts some wavelengths and cancels others. Tilt the feather and the spacing relative to the light changes, so turquoise becomes emerald, then bronze. Many beetles wear a similar secret, stacks of chitin reflectors that turn them into living metallic coins. The famous blue of a morpho butterfly wing comes not from a blue pigment, but from rows and ridges that act like a diffraction grating. They reflect and reinforce slices of the spectrum to produce a blue so saturated it can be seen from far away in the rainforest gloom. Hummingbirds go one step further. Tiny platelets in their feathers make colour that switches with their angle, so a gorget looks black until the bird turns and suddenly ignites. To a human eye it's a surprise. To a female hummingbird it's a sentence. As summer leans toward fall, trees run a quiet audit. Chlorophyll, the green pigment that funnels sunlight into chemical energy, is expensive to maintain and vulnerable to cold. Before leaves are shed, trees pull nitrogen and other valuables back into twigs and trunk. When chlorophyll breaks down, the supporting cast steps forward. Carotenoids, pigments that have been there all along, reveal golds and warm yellows. They broaden a leaf's light capture during the growing season and then paint the hillsides when green retires. In some species, fresh pigments called anthocyanins are made right at the end, staining leaves ruby and crimson. These may act like sunscreen for aging leaves, shielding delicate machinery while the last nutrients are reclaimed, or they may signal to insects that this tree is well defended. Cool nights and sunny days concentrate sugars in leaves and favour brighter reds. Warm, cloudy spells tilt the palette toward ochre and bronze. When the job is done, a layer of cells at the petiole weakens, abscission, and a leaf rides the air down, its carbon and minerals cued for return to soil. We see with brains more than with eyes. Colour is not only what wavelengths arrive, but which ones the mind decides to emphasise. Under a warm lamp, a white shirt still reads as white because your visual system quietly subtracts the lamp's yellow. Outside under blue sky, the same shirt still reads as white. This constancy is useful and occasionally tricked, as with viral photographs that half the world sees as blue and black and the other half as white and gold. Many animals carry different assumptions. Bees see ultraviolet and follow nectar guides painted in patterns beyond our range. Reindeer shift seasonal sensitivity so they can spot lichens against snow under the blue heavy light of Arctic winter. Mantis shrimps pack a startling variety of photoreceptors, tuned across the spectrum and into polarization, trading fine gradations of hue for speed in recognizing signals flashed by other mantis shrimps. A dragonfly's eye, crowded with ommatidia, can read polarized reflections off water to find a place to land. Color, here, is not decoration. It is a routing tool. The ocean looks blue because water prefers to absorb reds and let shorter, bluer wavelengths travel farther. Skies look blue for a different reason. Air molecules scatter short wavelengths more strongly than long ones, so blue light is bounced around the dome and into your eye from every direction. Sunsets lean red because the path through the atmosphere is longer at the horizon, and the blue has been scattered out of your line of sight. Blood looks red because hemoglobin's iron center interacts with light in ways that reflect the long wavelengths and absorb others, carry oxygen, and the shade shifts, making arterial blood bright, venous blood darker. Opals flash rainbows because their silica spheres are neatly arrayed, diffracting different wavelengths at different angles, nature's photonic crystal in a fingertip-sized pebble. Cephalopods, octopuses, cuttlefish and squid paint with physics and cells at once. Their skin houses chromatophores, tiny elastic sacs of pigment that expand or shrink to change overall colour in milliseconds. Beneath those sit iridophores and leucophores, structures that reflect and scatter light to create iridescence and chalky white. By combining pigment changes with structural reflection, a cuttlefish can match a mottled rock, then ripple a passing wave pattern across its body to confuse predators, then flash high contrast stripes for a courtship display. Chameleons, despite old myths, don't swap paints under their skin. They shift the spacing of guanine nanocrystals in the dermis, tuning structural colour like a dial. A slow calm chameleon reflects more green. An excited or dominant one changes the lattice and pushes reflection toward yellow and red. Physics does the heavy lifting. The animal cues the lattice. Flowers play both sides too. Many petals hold pigment molecules that absorb specific wavelengths, the reason a poppy is red or a daffodil is yellow, but they also carry nanoscale wrinkles and wax crystals that scatter light to produce a velvety look or a matte bloom. Some produce ultraviolet bullseyes invisible to us that guide pollinators to nectar like runway beacons. The blue of many flowers is especially interesting because true blue pigments are rare. Plants sometimes stack structural tricks with chemical ones to bend a purple anthocyanin toward blue by adjusting pH, adding metals like aluminum, or altering the cell sap's co-pigments. Hydrangeas famously shift from pink to blue depending on soil acidity and aluminum availability, a homeowner's science experiment in a front garden. In forests and oceans, colour often hides rather than advertises. Countershading makes animals darker on top and lighter below, so they appear flatter and less three-dimensional in ambient light. The same principle painters use with shading, reversed to erase volume. Some open water fish carry mirror-like sides made of guanine crystals that reflect whatever colour the sea happens to be, dissolving into their backdrop. Snow hares swap brown for white seasonally, a pigment relay cued by day length rather than temperature. A smart bet in places where the calendar is more reliable than the weather forecast. There is a practical rhythm to all these effects. Biological systems borrow the rules of optics and then tweak chemistry to suit. Pigments subtract, structures add and rearrange, heat is minimized when it would be wasteful, contrast is sharpened when it would be useful. Predators intercept the sensory assumptions of prey, flowers intercept the sensory assumptions of bees. The geometer and the biochemist share a lab bench. So when a wave breaks at night and throws sparks, you're watching a cell's emergency alarm rewritten as magic. When a peacock turns and the field goes quiet for a heartbeat, it is a demonstration of nanoscale carpentry. When a hillside lifts from green to gold to crimson, it is a tree closing its accounts and revealing a palette that was there all along. The colours of life are more often negotiations than facts, light bent by structure, filtered by molecules, and interpreted by eyes and minds tuned by evolution. The rock record reads like an old diary, and some of the entries are written by animals that hardly change their handwriting. Horseshoe crabs scuttle the shallows wearing armour that would not look out of place beside trilobites. Their basic plan, a domed carapace, spiked tail for righting themselves, rows of book gills and blue copper-based blood, has worked for roughly 150 million years. Living fossil is a clumsy phrase because nothing living stands still, but the outline endures because the design still solves the same problems. Breathe in thin water, weather waves, sift for worms and clams in the sand, grow by molting a shell and stepping out a size larger. If you want to glimpse an ancient shoreline, watch one at high tide on a spring night when they heap together to spawn. The scene has played out since continents wore different names. Travel deeper in time and the cast gets smaller, but the influence grows. Many of the bacteria tucked into rocks and soils today are lineal descendants of Earth's earliest residents, kin to the cells that learned to harvest energy from minerals, hydrogen and light, long before plants or animals existed. The deep biosphere, kilometers down, houses communities that eat the products of rock-water reactions, fueled by the slow rain of radiolytically split molecules and the steady chemistry of serpentinizing mantle. They divide on patient schedules, years, decades, yet they endure, proof that habitable includes places we cannot see. The last universal common ancestor, the ghost at the root of the tree of life, was almost certainly microbial, and much of what cells do now, copy DNA, translate RNA, harvest gradients across membranes, was already old when mountains we now climb were seafloor mud. The oldest widely accepted fossils do not look like animals at all. They're layered rocks called stromatolites, built a hair's breadth at a time by microbial mats. Picture a slimy carpet of cyanobacteria trapping silt, growing upward toward light, trapping more, then growing again until a dome forms. Slice through, and you find laminations like the pages of a book, each page a day or a season lived by communities that photosynthesized when the world was young. Modern stromatolites still grow in places like Shark Bay, Australia, where salinity keeps grazers thin and mats can survive. Their ancient cousins did more than pile stone, they changed the atmosphere. When cyanobacteria evolved the trick of splitting water and using the electrons to fix carbon, oxygen was the exhaust, At first it rusted iron out of the oceans, leaving banded iron formations like candy canes frozen in bedrock. Later, when the buffers were spent, oxygen began to accumulate in the air. That great oxidation, spreading over roughly 2 billion to 3 billion years ago, was both catastrophe and opportunity. For organisms not built for it, oxygen was a corrosive poison. For the rest, it opened a highway. Aerobic respiration, which yields far more ATP per molecule of fuel, became possible, and with it the chance to power bigger, more complex bodies. Ozone formed in the upper atmosphere, filtering ultraviolet radiation and softening the surface for life. Earth's rusted red rocks are signatures of that upheaval. Your hemoglobin, picking up and dropping off oxygen with tidy chemistry, is a beneficiary. The air you just pulled into your lungs has a lineage that runs back to sticky mats in tidal flats. When we tell the story of the evolution, we often dwell on the brief, showy chapters: the Cambrian explosion, when body plans diversified and teeth and claws complicated daily life, the march of vertebrates onto land, the reign and fall of dinosaurs. These make good pictures, but the timeline is mostly microbes. For billions of years, the planet was a microbial world with occasional experiments in multicellularity. Even after animals appear, microbes remain the engineers, fixing nitrogen into forms plants can use, decomposing the fallen into raw materials for the next growth, cycling sulfur, iron and manganese, filling vent fields and desert crusts and the skins and guts of the larger beings. The Burgess Shale and Changjiang fossils are glorious snapshots of an animal moment. The black smoker chimneys and microbialites are the steady film. The continuity shows up in tools as well as forms. The proton-powered turbine that makes ATP in your mitochondria is a refinement of pumps ancient bacteria used across their membranes. Photosystem machinery in chloroplasts traces to cyanobacteria that once free-floated in green water. Even the genetic code, the mapping from codons to amino acids, is overwhelmingly conserved, with only tiny edits at life's margins. This unity is why a jellyfish gene can make a mouse cell glow, and why bacteria can be coaxed to brew human insulin. The instructions are old, shared, and still legible across kingdoms. Of course, the deep past was not a steady climb. Icehouse episodes may have cloaked the planet in glaciers from pole to pole. Snowball Earth, with life riding out the cold in brine channels, volcanic oases, and patches of open water. Supercontinents assembled and tore, rerouting currents, revamping climate, knitting and unknitting habitats. Mass extinctions arrived like punctuation, volcanic outpourings, anoxic seas, an asteroid that darkened the sky. Through each break, microbial life persisted and reseeded the world. When the lights dimmed, chemotrophs, working on minerals, kept going. When the air soured, anaerobes shrugged. Resilience in the small bought time for novelty in the large. If you hold a piece of banded iron in your hand, red jasper, grey hematite, you're touching the breathing of oceans older than animals. If you run a finger across a stromatolite slab, you feel the rise and fall of mats that felt tides long before trees shaded coasts. On a summer night, if you watch a shore studded with helmeted crabs, you're witnessing an echo of a Paleozoic beach, as if time folded for an hour to let the old scene replay. None of it is a museum diorama, all of it is still connected to the present. The microbes that invented oxygenic photosynthesis lit the fuse for the metabolisms in your cells. The ancient chemistries that let cells harvest energy from iron and sulfur still run in mines, hot springs, and the belly of the earth. The old architectures, rings of DNA, rotary enzymes, lipid membranes, were good ideas and remain so. So much of what we call modern is inheritance with edits. Horseshoe crabs carry a plan that works, so they keep it with minor notes. Bacteria remember how to eat rock and light, so they do, and they teach the lesson to symbionts living inside plants and animals. Microbial Mats wrote the first long chapter of life's book, oxygenated the stage, then kept turning pages while larger actors took their turns in the spotlight. If you want to understand evolution's tempo, trust the small and the many. They set the key, they hold the rhythm, and when the melody modulates, as it always does, they are the ones who play through. You're home to a crowd with an enormous library. In the folds of the intestine, trillions of microbes carry a collective catalogue of genes that dwarfs your own. Millions of instructions for breaking down fibres, crafting vitamins, modifying bile acids, and simmering plant chemicals into new forms. By comparison, the human genome contains roughly 20,000 protein-coding genes. That imbalance is not a threat, it is leverage. When you eat a varied plant-rich meal, the menu is partly for you and partly for them. Microbes ferment fibres you cannot digest into short-chain fatty acids that feed the cells lining the colon, strengthen the gut barrier, and whisper to the immune system to stay calm but ready. They synthesize vitamin K and several B vitamins, trim and tweak hormones, and even nudge how the liver handles fats and sugars. Change the diet and the community shifts within days, like a city changing tempo when a festival comes to town. New stalls appear, old streets quiet. The overall mood rebalances. The more diverse the crowd, the more stable the whole, which is why a wide range of plants tends to support steadier digestion and a steadier mind. Step outside after a dry spell and the first raindrops uncork a scent most people know immediately. That smell of rain, petrichor, has a microbial signature. Soil-dwelling actinobacteria, especially a group called Streptomyces, spend their lives taking apart tough plant remains and building useful molecules. One of their byproducts is geosmin, an earthy compound so distinctive that human noses can detect it at astonishingly low concentrations, like a few drops in an Olympic swimming pool. During drought, geosmin accumulates in dry soil and in the pores of rocks. When rain arrives, the impact of droplets traps air and launches tiny aerosols into the air. Those bubbles carry geosmin and plant oils to your nose. The same chemistry may guide animals to water or to fresh soil. In a way, microbes are broadcasting the news. Moisture has returned. Life can resume its faster pace. Your immune system grew up in conversation with microbes, both the friendly ones you host and the invaders you fight. From birth onward, cells in the gut sample passing material and present bits of it to lymphocytes, teaching them what self, friend, and foe look like. Useful exposure strengthens a balanced response, tolerant to food and the normal microbiome, fierce toward pathogens. Certain bacterial metabolites, the short-chain fatty acids made from fibre, directly encourage regulatory T cells that keep inflammation from running away. The immune system's sensors, toll-like receptors and other pattern recognizers, didn't appear out of nowhere. They evolved to spot molecular signatures that many microbes share, like the lipopolysaccharides in some bacterial walls or fragments of flagellin. Those ancient alarms are still wired to your most modern defences. The deeper origin story is even more inventive. The adaptive immune system's ability to generate a vast repertoire of antibodies and T-cell receptors depends on a cut-and-paste trick. Gene segments are shuffled and stitched into new combinations in developing lymphocytes. The enzymes that do the cutting look like descendants of an old mobile genetic element, a wandering piece of DNA that learned to help its host instead of simply hopping around. From a molecular vagabond came the means to recognize almost any new threat. Even memory has roots in old battles. After an infection, some B cells and T cells linger as sentinels. Upon re-encounter, they flare into action faster and stronger. The idea, remember and respond, began as a response to microbes and now underlies vaccinations and much of public health. Your mitochondria carry reminders that the boundary between you and the microbial world is not a hard wall, but a negotiated line. These little powerhouses resemble bacteria in several ways. They have their own small circular genome. They divide by fission. Their ribosomes and membranes are bacterial-like, and their inner membrane carries cardiolipin, a signature lipid more common in bacterial cousins. They convert the energy in food into ATP by running electrons along a chain of protein complexes and using the payoff to pump protons, stockpiling them like water behind a dam. A rotary enzyme lets the protons flow back, minting ATP, useful energy coins for everything from muscle contraction to nerve impulses. Almost all of their proteins are now encoded by nuclear genes and shipped in. But the remnants of independence remain, including the peculiar inheritance pattern. Mitochondrial DNA, with rare exceptions, is passed down the maternal line. That ancestry still echoes in the way the immune system reads cell damage. When mitochondria are torn open by injury, they release molecules, N-formyl peptides, fragments of their DNA, that the body interprets as bacterial danger signals. Immune cells rally as they should, because for most of life's history those signals meant invader. In the heart and brain, which depend on steady mitochondrial work, anything that knocks these organelles off balance can ripple outward. Fatigue, muddled thinking, slowed healing. Training and sleep coax mitochondria to multiply and streamline. Chronic stress and inactivity let them idle. The partnership responds to how you live. The microcosm reaches the brain by multiple roads. The vagus nerve provides a direct hotline from gut to head. Immune messengers cross from blood to brain. Microbial metabolites influence the production and sensitivity of neurotransmitters like serotonin and GABA. After a week of disrupted sleep or a course of broad-spectrum antibiotics, it is common to feel a little out of tune. Appetite odd, patience thin, focus scattered. Reintroduce diversity in food, let the daily rhythm settle, and the internal weather clears as the community inside finds its footing again. Fermented foods offer friendly visitors. Fibre offers steady groceries. Time does the rest. Antibiotics remain among medicine's greatest tools, and using them wisely means remembering the ecosystem they affect. A drug aimed at a dangerous bacterium also thins the ranks of helpful neighbours, leaving niches for opportunists to bloom. Most communities recover with time and diet, but during the reset, digestion and immunity can wobble. Precision helps, narrow drugs when possible, unnecessary courses avoided, and after treatment, a return to varied foods that feed a wide range of residents. Think of it as reseeding a meadow after a fire. Even senses outside the gut carry microbial fingerprints. The mouth is a mosaic of neighbourhoods, tongue, cheeks, teeth, each with its own biofilms. When the balance tilts, acids from sugar-loving bacteria etch enamel and gums inflame. When it steadies, saliva and beneficial species buffer the chemistry and keep the peace. Skin hosts shift with moisture and oil. Routine washing prunes excess without scouring the community to bare rock. Everywhere the pattern repeats. A well-fed, well-balanced microbiome supports the tissue that houses it. So the human body is not a lone creature but cooperative, a composite of human cells, microbial partners, and endosymbiont powerhouses, all speaking chemistry. The smell of first rain, the calm after a nourishing meal, the stamina that grows when you move and sleep well, even the energy minted in each heartbeat, all carry signatures of ancient arrangements. You are more than one genome. You are a habitat, a history, and a set of alliances that make you possible. Intelligence does not always arrive in a skull. Sometimes it creeps as a yellow sheet across a petri dish, splitting, retracting, and trying again until the problem is solved. Slime moulds, especially the star of the show Physarum, are single-celled giants, living blobs with thousands of nuclei spread through a single membrane. They move by shuttling cytoplasm back and forth in slow pulses, like tides on a table. Put oat flakes at the exits of a maze and let the slime mould loose at the entrance. At first it spreads everywhere, sampling, laying down a network of tubes. Where food signals are strong, tubes thicken. Where they are weak, tubes dwindle. After a few hours, the mess reduces to a single efficient path linking start to feast, the shortest route, found without a blueprint or a brain. Change the position of food and the network rewires, reinforcing new roads and pruning the old. It's not thinking in the way a person thinks. It's a feedback loop between chemistry and shape. Sense, grow, feel the improvement, grow more there, abandon the rest. The same trick scales up. Arrange oat flakes to mimic a map of cities, and Viserum will weave a rail system that looks suspiciously like what human engineers built. Redundant loops to hedge against brakes, fat highways for high traffic, skinny spurs for side towns. The organism is solving a difficult optimization problem by existing. It uses local rules, follow gradients of sugar and moisture, retreat from salt and light, and the global pattern emerges. Even memory in a slime mould sort of way appears. If the blob crosses a patch it doesn't like, such as a mild salt barrier, its rhythmic pulses slow and its tubes constrict. Come back later and it crosses faster, as though the unpleasantness were logged. The logbook is not in neurons. It's in the altered viscosity of the cytoplasm and the geometry of the tubes. The body is the memory. Plants write their decisions in slow motion, mostly underground where eyes rarely look. A root tip is capped with a helmet of cells that sense gravity by letting tiny starch granules settle. That's how down is known. But gravity is just the background. Roots also taste and smell. Nitrate and phosphate tug growth their way. Toxic metals push growth aside. Strands lean toward moisture hidden in drier soil, picking the faint trace of water moving through sand. In crowded beds, roots can even distinguish kin from strangers and reduce competition with their own relatives. The currency of this decision-making is hormone flow. Auxin pools on one side of the root to slow cell expansion there, which bends the tip in the other direction. Ethylene whispers, "Watch out," when oxygen is scarce. Cytokinins and abscisic acid adjust priorities when drought or salt intrude. Electrical spikes. Yes, plants have them. Race along the root in response to wounds or sudden changes, modulating ion channels and steering the growth front. No brain issues commands. Local rules and local measurements steer a living drill that threads through rock crumbs to exactly where resources lie. Above ground, similar judgments unfold in leaves and stems. Stomata open to drink in carbon dioxide but slam partway shut if the air turns desert dry. Tendrils sweep slow circles until a touch signal says, "Anchor here," then tighten into perfect coils. Pollen tubes, single cells on an urgent mission, navigate down a stile, rerouting around obstacles, reading chemical signposts that the ovule emits to home in on the target. Each choice is small. Open or close, thicken or thin, turn or continue. A million small choices paint the larger picture we call growth. Fungi take the maze-solving spirit and bury it in the world. A fungal mycelium, white threads thinner than hair, fans through soil and rotting wood, sampling, branching and fusing into a net. Where nutrients are rich, hyphae thicken. Where pickings are poor, they pull back. Pipes link into loops that maintain flow even if a segment is damaged. Sugars delivered from a partnering plant arrive at some nodes. Elsewhere, nitrogen or phosphorus is abundant. The network balances supply and demand by redirecting traffic. When a log runs dry, the fungus shuts valves and invests in a different vein. Studies that measure nutrient flow find that mycelia behave like frugal engineers, minimizing transport costs while keeping reliable routes, exactly the balance human planners try to strike with roads and fibre optics. There is no headquarters in the dirt issuing orders. The logic lives in the plumbing. Even bacteria, the smallest of soloists, tally votes. In a puddle of warm broth, each cell leaks tiny molecules into the water. As the crowd grows, the concentration rises. When it crosses a threshold, the population flips behaviour together. Harmless loners shift to communal film builders. Glowing species switch their light on at once. Pathogens activate attack genes only when there are enough comrades to overwhelm a host's defences. The habit is called quorum sensing, and it is politics in a droplet. Count noses, then act. Sunset waves of bioluminescence across reefs and synchronized shifts inside biofilms both spring from that microscopic census. What ties all these examples together is computation without a central processor. Information is gathered locally and stored physically, in thicker tubes, in a route's new direction, in a patch of mycelium that was reinforced yesterday. Signals are analog, more or less hormone here, more or less nutrient there, and the body behaves like a spreadsheet that recalculates with each new cell added. If a section is cut away, the rest reconfigures without panic. If conditions change, thresholds slide and new patterns firm up. This kind of intelligence is robust because it is distributed. There is no single point of failure, no fragile master plan. A slime mould can be chopped, and each piece continues the algorithm. Plant roots find detours around stones. Fungi route around a boot print as gracefully as a city routes around a closed bridge. The lesson is not that neurons are overrated. Brains excel at compressing space and time, at rehearsal and imagination. But life's broader lesson is that decision-making can arise from the structure of living material itself. Geometry couples to chemistry. Feedbacks emerge, and suddenly a creature without nerves solves a puzzle a student might tackle on paper. The rules are simple. Sense the gradient. Reinforce success. Abandon failure. Leave a trace that biases the next choice. If you build a body that can follow those rules, intelligence becomes a property of motion and matter, not just of grey tissue. Watch a time-lapse of a slime mould and you'll see a neon map of choices settling into an efficient truth. Peek under a stone and a root will be threading a contour line to a hidden pocket of water. Scrape a rotting log, and a fungal fan will be redrawing itself to match the day's accounting. None of these systems has a face to look wise, but each carries a quiet competence. Life, it turns out, does not need a brain to know what to do next. Even the quietest garden keeps time. Many plants fold their leaves at dusk and reopen them at dawn, a slow-motion bow tied to an internal clock. This circadian rhythm ticks along even in constant darkness for days, proof that the timing is set within the plant rather than pulled entirely by the sun. In leaflets and flowers, tiny hinge-like structures swell or shrink as ions move in and out, shifting water and pressure to open and close. Stomata, the microscopic pores that let carbon dioxide in, tighten overnight to save water and reopen with first light. Chloroplasts rearrange inside cells to catch morning rays and retreat from the harshest noon glare. Genes, too, cycle. One set turns on at dawn to prime photosynthesis, another peaks at night to handle repair and resource shuffling. External cues like light and temperature fine-tune the schedule, but the beat is set by a core loop of clock genes that turn one another on and off in a steady day-length cadence. Plants do not dream, but they do keep appointments with the day. Bodies keep similar appointments, and in sleep they make use of the dark hours for maintenance we barely notice. DNA suffers nicks and chemical scuffs during waking life from ordinary metabolism and stray radiation. At night, repair crews get more road space. Enzymes patrol the genome, cutting out damaged bases and knitting strands back together. Cells slow non-essential tasks, freeing energy for upkeep. The brain, which rarely goes off duty, uses sleep to run a deep clean. Cerebrospinal fluid pulses through channels that widen at night, washing away metabolic byproducts like beta-amyloid and tau that, left to accumulate, are linked to cognitive decline. This glymphatic flow is most vigorous in deep non-REM sleep, when neurons settle into slow, synchronized rhythms and the tissue's electrical chatter quiets enough for plumbing to take the stage. Hormones keep time with this maintenance. Darkness nudges the pineal gland to release melatonin, a signal to tissues that night has begun. Growth hormone pulses shortly after you fall asleep, cueing protein synthesis, tissue repair, and the strengthening of bone. In the immune system, cells that coordinate defense patrol and proliferate more efficiently during sleep, and inflammation is tuned down from a simmer that, if left high, wears the body thin. Muscles benefit as microtears from daytime effort are stitched. Glycogen stores are topped up, and mitochondria, the cell's powerhouses, adjust their numbers and tune their engines for tomorrow's demands. Even skin joins the shift. Blood flow to the surface rises slightly, water loss increases, and repair of sun-induced damage accelerates. Morning light then lands on the retina and starts the counter timer for the next melatonin rise, resetting the 24-hour loop. Memory does not wait for morning to settle in. It is edited and saved while you sleep. During non-REM sleep, the hippocampus, the brain's rapid learning notepad, replays patterns of activity that occurred during the day. In miniature rehearsals called sharp wave ripples, sequences of neurons fire in the same order they did when you learned a route, practiced a chord, or studied a face. These bursts align with waxing and waning waves in the cortex, and synapses strengthen along the relevant pathways, moving fragile impressions toward longer-term storage. In REM sleep, when the eyes dart beneath closed lids and the brain's activity looks surprisingly wake-like, emotional memories are integrated and associations are explored, often in the surreal stagecraft of dreams. The balance between non-REM and REM across the night lets the brain both prune and fortify. Some connections are dialed down to reduce noise and energy cost. Others are bolstered to capture what matters. This nightly pruning is as important as the strengthening. Throughout the day, synapses tend to ratchet upward. It is how learning accrues. But all the time would be metabolically expensive and would drown signals in static. Sleep provides a gentle reset synaptic homeostasis that lowers overall synaptic strength a notch while preserving the relative differences you earned with practice. The next day starts with headroom for new learning, like a chalkboard wiped clean except for the important lines. People notice it as clarity after a good night's rest, or fog after a fractured one. The same brain, but with the books either shelved or still in piles. Rest reaches beyond the brain. The heart enjoys lower blood pressure and heart rate overnight, a relief valve that daily helps protect vessel linings. The liver shifts gears to handle detoxification and to balance blood sugar for the fast ahead, breaking down or building up stores as needed. The gut, done with major digestion, runs housekeeping waves that sweep residual contents forward, a quiet conveyor belt that tends to stall when sleep is too short or too erratic. Even the microbiome keeps hours. Certain gut microbes bloom at night, metabolizing fibers differently than their daytime neighbors, and producing short-chain fatty acids that whisper to immune cells in the liver about the state of affairs. The clock that coordinates these chores sits in the hypothalamus, in a cluster of neurons that keep roughly 24-hour time by cycling their own genes. Light entering the eye trains this master clock. Food timing, social cues, and activity polish the edges. Peripheral clocks in almost every organ then sync to the conductor, each keeping its own variation on the theme. shift the light-dark cycle abruptly, flying across time zones for instance, and the parts desynchronize temporarily. You feel it as jet lag, hunger at odd hours, sleep that arrives early or not at all, a mind that wakes on a different schedule than digestion or mood. Gentle exposure to morning light, consistent meal times, and regular movement escort the clocks back into harmony. Plants, too, use the night for repair and rerouting. Without photosynthesis to power the day's manufacturing, they tap stored starch, metering it out so the reserve lasts until dawn, a quiet calculation that scales with the length of the night. Genes that handle oxidative damage and protein folding peak in darkness, mending the wear from sun and wind. Some flowers hold pollen closed overnight, guarding it from dew and nocturnal grazers. Others open only in the dark to meet moths and bats, timing scent release like a bakery's morning batches. The rhythm is not sleep as animals know it, but it is rest, reduced responsiveness, repeated daily, with repair and energy balance at its heart. Not all rest unfolds in one long stretch. Within the day run shorter cycles, ultradian rhythms of about 90 minutes. Attention rises, plateaus and dips. Performance on complex tasks follows the curve. Respecting those waves, working in focused bouts and taking brief, genuine breaks, mirrors what sleep does at night. Concentrate effort, then clear byproducts, then resume. Many animals partition rest even more dramatically. Birds and marine mammals can sleep with one brain hemisphere at a time, keeping an eye out while still logging maintenance. Some small mammals enter torpor, a controlled, temporary lowering of body temperature and metabolism that saves fuel when food is scarce or nights are bitter. Across strategies, the theme is conserved. Step back to preserve the instrument. Calling rest idleness misunderstands the accounting. A field left fallow replenishes. A muscle between sets grows stronger. A mind that wanders after study is often binding ideas in the background. Sleep stitches up the day's small tears, files the new, discards the redundant, and sets the stage for tomorrow's work. Plants fold their leaves, cells mend their letters, neurons hum a quieter song, and fluids wash the circuitry. The rhythm is older than flowers and bones, held by microbes that cycle with tides and temperature, and by the clocks in green stems and grey matter alike. To follow it is not indulgence, it is to keep time with life's most reliable metronome. In the coldest seas, the danger isn't merely brewer, it's physics. Seawater can fall below zero without freezing solid because salt lowers its freezing point. A fish's blood, being less salty, should freeze first, ice forming inside vessels like frost on a window. Some polar fishes dodge that fate with antifreeze proteins. These small, exquisitely shaped molecules coat the surfaces of tiny ice crystals and stop them from growing, a trick called thermal hysteresis. Instead of letting crystals add layer upon layer, the protein bends the crystal surface into a shape that is energetically unfavourable for more water molecules to join. The result is not warmer blood, but safer blood. Ice can't take hold. Different lineages arrived at the solution independently. Antarctic notothenioids and Arctic cods both make antifreezes, proof that evolution will rediscover a good idea when the environment demands it. There's a cost. Manufacturing proteins is expensive, and the same sticky qualities that bind ice can sometimes snag other molecules. But in water cold enough to flash freeze an unprotected fish, the investment pays out every second. Cold survival comes with a second challenge. Oxygen moves sluggishly in chilled water, and chemical reactions slow. Polar fishes tune their whole metabolism downward, swapping speed for stability. Enzymes evolve flexible hinges so they can still fold and catalyze at low temperatures. Membranes become richer in unsaturated fats to stay fluid. the biochemical equivalent of adding winter-grade oil to an engine. Some Antarctic species even lack red blood cells and hemoglobin altogether, relying on the greater solubility of oxygen in icy water and on unusually big hearts and wide blood vessels to push enough O2 around. Viewed up close, the solution is a bundle of compromises: antifreeze proteins to bar the door to ice, slippery membranes for winter motion, and circulatory plumbing scaled for syrupy conditions. None of this makes a sprinting predator. It makes a survivor that treats cold like a slow river. It has learned to swim. At the other extreme, heat is not the main villain of deserts, thirst is. Many desert plants meet it with a decision that looks like magic. They stop. Resurrection plants, such as Selaginella lepidophylla, curl into tight brown balls during drought, losing almost all free water from their tissues. Metabolism drops to a whisper. Inside, sugars like trehalose and raffinose replace water around proteins and membranes, forming a glassy matrix that keeps structures from collapsing or fusing while dry. Special LEA proteins act like molecular cushions, preventing the crumpling that would normally kill cells. Antioxidants stand guard to soak up reactive oxygen that builds when electron flows stall. When rain finally returns, hours, months, even years later, the plant drinks, the glass softens, enzymes find their shape, and the frond unfurls green as if nothing happened. It is not resurrection in the mystical sense. It is superb materials science, trading motion for preservation, and then reversing the choice on cue. Desert annuals run a parallel strategy by outsourcing survival to time capsules. Seeds with armoured coats and chemical inhibitors lie buried, durable enough to ignore a dozen teasing drizzles. Only a soaking rain cracks the seals and leaches inhibitors away. Germination then races. Roots punch into moist soil, leaves unfurl, flowers open, seeds set, and the next generation drops back into the bank before the ground crusts and the air sizzles again. Within a population, seeds vary in how much convincing they require. Some germinate on the first good soaking, some wait for the second or third season. That built-in bet hedging keeps the lineage alive through strings of bad years. The plant's decision is distributed across many seeds and many springs, a silent arithmetic of risk. Cold deserts do the same arithmetic, only slower. Arctic and Antarctic mosses have been coaxed back to life after centuries entombed in ice. While frozen, their cells endure in a vitrified state similar to the glassy preservation of resurrection plants. DNA repair enzymes reawaken with meltwater and begin fixing damage accumulated from background radiation and time. Chloroplasts dormant in the dark restart photosynthesis within days of light returning. These mosses rebuild from tiny living tips as if a long pause were only a deep breath. In permafrost soils, microbes revive after thousands of years, as do some hardy nematodes. In alpine scree, lichens resume growth after years beneath snow. Life, it turns out, can treat time like an accordion, stretch when resources are rich, compress when they are not, and still carry the tune. Animals in parched places borrow the playbook too. Tardigrades and some desert insects enter cryptobiosis, a ton-like state where metabolic rates become nearly immeasurable and protective sugars vitrify the interior. Brine shrimp eggs, sold as sea monkeys, can be dried and stored for years. Add water, and a cloud of swimmers hatches from what looked like dust. Even vertebrates flirt with time control. Some desert frogs cocoon themselves in layers of shed skin, burrow deep, and wait through multi-year droughts with a slowed heartbeat and a metabolism fueled by stored lipids. First, heavy rains dissolve the cocoon and wake the frog to a sudden singing world. Across all these strategies, a pattern emerges. Survival often means controlling phase changes, water to glassy matrices, ice to constrained crystals, liquid to vapor, and trimming metabolism until chemistry can be trusted again. The tools are surprisingly universal. Sugars stabilize fragile shapes. Chaperone proteins keep neighbors from sticking. Membranes are tuned with unsaturated fat so they bend without breaking. Enzymes swap rigid bonds for springier ones so they can still move at the edge of freezing. DNA repair systems stay on retainer, ready to mend the slow scuffs of time the instant energy returns. The systems do not deny physics, they negotiate with it. There's a certain humility in all of this. When conditions turn hostile, the winning move isn't heroism, it's restraint. Fish accept slower lives to keep blood liquid. Desert plants choose stillness over waste, knitting a glass inside their cells and waiting for a real rain. Arctic mosses trust that spring will eventually honour its appointment, even if it's centuries late. Seen from a distance, these are stories about temperature and water. Up close, they are stories about pacing, about living things that learn to stretch a moment until better moments arrive. Slowing time is not an evasion, it is an adaptation, a way of keeping the thread of life unbroken as seasons, climates, and ages pass overhead. The end of a living thing is not an erasure, it is a handoff. When a leaf falls, when a beetle quiets, when a tree comes down in a storm, a different crowd steps forward and begins to take it apart with exquisite patience. Decomposition is not rot as failure. It is a choreography of eaters, each with a specialty, turning bodies back into ingredients. First come the swift opportunists, flies laying eggs, beetles prying seams, microbes waking to a sudden feast. Then arrive the larger cleaners. Carrion birds and mammals strip muscle and sinew, scattering what they cannot finish. Beneath, a city you rarely see goes to work. Springtails, mites, millipedes, woodlice, and earthworms shred what remains into smaller and smaller pieces, opening surface area for the finest artisans of decay, bacteria and fungi, to complete the chemistry. Fungi are the headline act because they wield tools no one else does. Their hyphae, those pale threads that push through wood and leaf, oozes enzymes capable of unlocking lignin, the tough, complex polymer that gives wood its resistance. Where most creatures gnaw at the edges, fungi unbraid the knot itself. White rot fungi can crack lignin's aromatic rings, leaving pale, ghostly wood behind. Brown rot fungi take the cellulose and hemicellulose, cubing the timber into crumbling blocks and letting rain finish the job. In both cases, what was once a trunk becomes a fine mix of mineral and organic particles we call soil. Along the way, fungal glues knit crumbs together into stable aggregates that hold water and air, making room for roots and microbes. The forest's future stands on the scaffold of its past. Bacteria handle the quick, soluble work, sugars, proteins, and simple fats. They multiply fast in the moist pockets that decomposers create, turning easily digested molecules into carbon dioxide, water, and a suite of small acids and gases that other creatures can use. Together, bacteria and fungi run assembly lines that reclaim nitrogen from amino acids and chlorophyll, returning it to forms plants can take up through roots. They free phosphorus from cell membranes and ATP, release sulfur from proteins, and liberate trace metals needed by enzymes across the food web. When conditions are oxygen poor, deep inside a waterlogged log or in a swamp, different crews take over, breathing nitrate, sulfate, or even iron instead of oxygen. The end products change, but the principle does not. Elements are passed from form to form, not lost. Detritivaries, the shredders, are the quiet accelerators. A millipede's rasping mouth parts and a woodloose's steady grinding convert dry leaves and twigs into damp flecks where microbes can gain a foothold. An earthworm pulls litter into its burrow, digests what it can, and returns the rest as castings richer, in plant-available nutrients. Burrows channel rain into the profile and let roots follow. In grasslands, dung beetles roll and bury, seeding the next season's green with every ball they stash. Nothing in this guild is glamorous, but their collective work keeps nutrients cycling quickly enough that life above ground can grow dense and bright. On a larger stage, scavengers close loops that would otherwise waste energy. Vultures, immune to many pathogens, sanitize landscapes by consuming carrion that could spread disease if left to fester. Hyenas and foxes finish what larger predators begin, and the leftovers still feed a chain of ever smaller mouths until almost nothing recognizable remains. Even in the sea, where a dead whale falls to darkness, the sequence repeats in a grander register. Sharks and hagfish take the soft tissues. Months later, the bones themselves host communities of worms, clams, and bacteria that dine on the fats locked within. A whale fall becomes an island of life for years, then fades as quietly as it arrived, its nutrients folded into the abyssal plain. Carbon rides this carousel in familiar costumes. In life it is sugars and starches, fats and flesh, cellulose and lignin. In decay, most of it returns to the air as carbon dioxide and in some anaerobic pockets as methane. Plants will pull it down again with light, rebuild leaves and stems, feed roots and microbes, and so the loop closes. Nitrogen cycles with a different beat. Decomposers release it as ammonium. Other bacteria oxidize it to nitrite and then nitrate. Plant roots sip these forms back into protein. When oxygen is scarce, denitrifying bacteria send some nitrogen back to the sky as N2, completing a loop that began when other microbes fixed that very gas into amino acids in the first place. Phosphorus and potassium move more slowly, banking in soils, riding in bones and seeds, washing in gentle trickles to streams and coasts, where plankton will use them and begin again. None of this diminishes the gravity of death. It reframes it. A tree's fall opens a hole in the canopy so light reaches the floor. Pioneer species flourish. Insects and birds arrive to harvest the new bounty. And beneath the scatter, the old trunk becomes a nurse log that cradles seedlings in a moist fungal-rich bed. A fallen animal becomes a pulse of fertility. Grasses flush greener where it lay, and those grasses feed herds months later. Even the materials of individuality become common goods. The calcium from bones stiffens eggshells and snail houses. The iron from hemoglobin finds its way into enamel and chlorophyll cofactors. The phosphorus in DNA winds up in new chromosomes that will copy and recombine. Your own body is written into this account. The water will evaporate to clouds and return as rain. The carbon will breathe back to the air and into leaves, apples and wheat. The nitrogen will join the proteins of roots, and then the muscles of deer or the seeds of a sunflower. The minerals, calcium, magnesium, trace metals, will settle into soil, taken up in slow, quiet sips by fungi-laced roots. Loved ones remember you in stories. The world remembers you in chemistry. Neither is trivial. Fungi's role as alchemists extends into partnership, not just cleanup. Many of the same mycorrhizal threads that digest wood plug directly into plant roots, trading minerals for sugar. As they dismantle a stump, they simultaneously ferry phosphorus to a sapling a few meters away. What looks like an ending is also a redistribution. Energy and matter move through a network, and the network decides, by the physics of flow and the biology of need, where to send the proceeds. In drought, some of that flow is water-wicked along high-full highways. In spring, it is nitrogen and phosphorus. In autumn, it is carbon stored as starch underground, a hedge against the coming cold. Decomposition has its seasons and its voices. In warm, wet weather, everything speeds. Fruit disappears overnight, leaves melt into dark tea, and the air smells of humus and spice. In cold or dry spells, metabolism slows, bodies persist. The ledger keeps its numbers but stops turning pages so quickly. Fire can shortcut the slow chemistry, volatilizing some nutrients, fixing others into ash that dissolves in the next rain. Afterward, fungi and bacteria step onto the fresh canvas, and succession begins anew. Even bones left on a desert crust will soften under dew and frost, surrendering their calcium to lichens and then to the firmer grip of roots. Nothing truly disappears is not sentiment, it is law. Matter changes form, energy flows and dissipates, fueling the work of transformation. Life uses the flow to build improbable order, trees from air, shells from seawater, thoughts from sugars, then returns the parts when the pattern ends. Death in this light is a change of state, a turning of one kind of order into the conditions for many others. It is not neat, and it is not immediate, but it is reliable and in a deep way generous. Walk a woodland path and you can read the ledger, a mossed stump feeding a ring of saplings, a scatter of feathers that became fox and fly, a rich black smear of soil that was leaf a season ago, the sweet cold scent after rain, as microbes breathe and everything briefly quickens. This is the balance, loss underwriting growth, endings underwriting arrivals, kept by armies of small workers who never take a bow. The catalogue of life is not finished, it is barely sketched. Each year, scientists add thousands of species to the list, and most of them are small, quick, and easily missed. The kind of lives that slip through sieves and seasons. A vial of seawater holds a galaxy of plankton that nobody has named. A scoop of soil contains microbes new to science. A moth flashing for a single week on one hillside can be discovered and lost again before another summer. DNA barcodes and environmental DNA widen the lens. A litre of river water can reveal the ghosts of fish, frogs and insects that passed hours or days ago. A roll call written in stray cells. Satellites spot unfamiliar forests pulsing with unusual greenness. Acoustics pick up bird dialects no one had recorded. Machine vision notices patterns our eyes glide past. The frontier is not a line on a map but a resolution knob. Turn it, and the world gains detail. While discovery continues, we're also gingerly joining the editor's room. Gene editing, first with meganucleuses and zinc fingers, then the elegant scissors of CRISPR, gives researchers a way to change a letter in DNA rather than waiting for chance. Base editors swap one nucleotide for another without cutting the strand at all. Prime editors can write small new phrases into the script. In medicine, the promise is sobering and precise. Fix the spelling mistake that breaks a blood cell's oxygen-carrying protein. Disable a receptor a virus uses as a doorknob. Tune immune cells to find and remember cancers more effectively. In agriculture, a leaf can be taught to tolerate heat, a root to find phosphorus more efficiently, a grain to resist a rust that has learned to hop continents. None of this is magic. Biology bills in trade-offs. A sturdier plant may pay in slower growth. An edited cell must be checked for unintended edits. Ethics are not an accessory, they are the harness and brakes. But the tool set is in hand, and with it the weight of choosing when to use it. Beyond editing existing lines, synthetic biology asks a bolder question. What if we build? Minimal cells strip genomes to essentials to see what life truly needs. Cell-free systems take the core machinery, ribosomes, enzymes, energy chemistry, out of cells and run it on a bench, a breadboard for biology. Designers stitch together circuits that sense a pollutant and respond by glowing, or that count cell divisions and then stop, like a self-expiring timer. Some labs introduce new letters beyond the familiar 4, expanding the alphabet so proteins can be made with entirely new building blocks. Others craft protocells, droplets wrapped in membranes that can grow, divide, and exchange molecules, clumsy cousins of cells that nonetheless teach us how compartments become chemistries and chemistries become behaviours. The first computer was not a laptop, it was a table of gears. Our first synthetic lives will be gear tables too, limited, fascinating, and more instructive than they are powerful. The edges of a live blur as we work there. Viruses carry genes and evolve, but they do not metabolize on their own. They borrow the host's factory. Prions are misfolded proteins that persuade neighbours to misfold. A terrible chain reaction that spreads information without DNA at all. Self-assembling RNA molecules can copy parts of themselves and mutate in a test tube, inching toward the simplest definition of evolution. Engineered phages sit between predator and payload, tools that can edit bacteria in hospital drains or in soil without touching anything else. Even inside us, mitochondria remain a domesticated other, with their own small genome and habits inherited from a bacterial past. The old schoolroom line, life begins here, ends there, starts to look less like a border and more like a fog bank, lifting and settling with context. Computation has entered the lab as collaborator rather than spectator. Protein structures that once took months to guess can now be predicted from sequence with startling accuracy, turning imagination into candidates in days. Models propose enzymes that bind a substrate nature never met. Experimentalists build them, fail, refine, and sometimes succeed. Whole genomes can be searched for small motifs that act like promoters and switches. Synthetic ones can be generated to order. But models inherit our biases. They are fast at drawing straight lines through the parts of biology we already understand, less reliable in swamps and cliffs. The savviest use is not to believe them, but to let them narrow a vast landscape to a manageable hike. With new powers come new responsibilities that cannot be shrugged off. A cheap edit can spread through wild populations if it confers an advantage, or if a gene drive is used to bias inheritance. That may sound like a way to rid islands of invasive rats or mosquitoes that carry fevers, but ecosystems are interlocking puzzles, and moving one piece can tilt the table. Guardrails are already part of the craft. Containment genes that require lab-only nutrients, kill switches that trip when an organism leaves its designed niche, ecological trials that begin in microcosms, then greenhouses, and only then, maybe, a cautious field. The long-term work is less technical and more civic. Governance that is open, local voices that can say no, benefits shared rather than hoarded, humility baked into timelines. Discovery and design will also be asked to serve repair. Coral nurseries crossbreed heat-tolerant strains, assisted gene flow ferries resilient genetics from warm reefs to cooler ones before heat waves arrive. Forests are replanted not as rows but as mosaics matched to future climates rather than the past. Soil, half of it living, all of it finite, is restored by feeding the quiet engineers. Cover crops for microbes, reduced tillage for fungi, messy edges for insects. A microbe taught to sequester more carbon in root exudates can serve as an unseen mason, mortaring carbon into aggregates that resist erosion. None of these solutions stand alone. They are old tricks. Cooperation, diversity, timing made deliberate. Meanwhile, the census of life will keep surprising us. In caves that exhale sulfur, pale insects have evolved on diets that would kill their relatives. In clouds, bacteria gather ice around themselves and seed rain. In the deep, microbes eat rock and store energy for what looks like forever, a patience that makes time feel elastic. New species will not all fit the romance of new tiger. Many will be bacteria known by their DNA before anyone sees their faces. Many will be ephemeral hybrids. Many will be lineages rescued from a last valley by a change in grazing or fire. The work of naming them is not trivia, it is jurisdiction. You cannot save what you cannot point to. Amid this expansion, the lists, the edits, the prototypes, the oldest rule still governs. Biology is a ledger of energy made orderly. Sunlight, chemical gradients, and a few lucky catalysts turn randomness into patterns, membranes that separate, cycles that repeat, sequences that copy, communities that trade. Patterns hold for a while, a cell, a leaf, a reef, then soften back into ingredients that can begin again. Whether we discover 1,000 new microbes next year, or rewrite 10 letters in a clinical trial, the accounting stays the same. Life borrows, arranges, spends, and returns. The future will likely look less like a single invention and more like a new kind of literacy. Children who grow up reading genomes as easily as roadmaps may choose to mend a gene the way we patch software, to restore a wetland the way we debug a network, to taste rain and hear in it the streptomyces and the dust it rode in on. The line between natural and synthetic will be drawn, redrawn, and then sketched lightly as we learn that all living things, including our inventions, answer to the same thermodynamics and the same ecological arithmetic. The stories we tell about life will change, more plural, more provisional, but their last paragraph will stay familiar. Energy becoming pattern, pattern becoming life, and life, after a long and busy while, becoming energy again. Thank you for drifting through this little night journey with me. If you enjoyed the gentle wander, if it helped you unwind, sparked a question, or nudged a sleepy smile, tap that like button so I know to make more. If you're new here, I'd love to have you in our cozy corner. Please subscribe to The Sleepy Scientist and turn on the notifications bell so you don't miss the next bedtime expedition. And to those already subscribed, you're the quiet current that keeps this channel moving. Thank you for listening, learning, and dreaming along with me. If something tonight surprised you, or you have a favourite fact you want me to dig into, leave a comment and I'll add it to my late night notebook. Until then, breathe deep, let the day soften at the edges, and let your thoughts settle like silt in clear water. Sleep well, take care of your curious mind, and I'll see you in the next one.